wise counselor, our perfect teacher, and our faithful friend. Thank you for allowing us today to meet and share our knowledge and time with one another in this seminar. May you extend your divine wisdom to our speaker so that he would be able to impart effectively his God-given knowledge to all of us. May he be blessed as he continues to bring his expertise to people who need them. Bless the participants as well, so that they would be able to learn the vital information from this activity. May you bestow your blessings after this seminar so that we may go out and spread what we learned in the spirit of your love and generosity. May we realize that this activity should glorify your name. Amen.
Welcome address by Dr. Mrs. Anita Kannagi, Organizing Secretary, Head of the Department of Zoology and Associate Professor, Jaira Jannabakyam College, Periyakulam.
Good morning to all. It is a glorious moment to extend my warm welcome on behalf of PG and Research Center of Zoology, Jaira Jannabakyam College for Women, Autonomous, Periyakulam, to the participants of the two-day international webinar on recent trends in life sciences. I convey my heartfelt gratitude to Dr. Nallasivam Palaniswamy, Associate Professor, Michigan Center for Translational Pathology, Department of Pathology, University of Michigan, and Associate Scientist, Department of Urology, Vaticuti Urology Institute, Henry Ford Health System. He did his undergraduate and postgraduate degree in the University of Madras, Chennai. Since he was much interested in human genetics, he completed his MPhil degree in the year 1984 and PhD degree in human genetics in the year 1993 in the University of Madras, Chennai. He worked as a postdoctoral research associate in molecular biology in the University of Vermont, Burlington, USA in 1990 to 1991, in Miami Children's Hospital Research Institute from 1993 to 1996, and as postdoctoral research associate in cancer cytogenetics in Memorial Sloan Kettering Cancer Center, New York, USA from 1996 to 1999. He was an approved licensor for the Certificate of Qualification for Laboratory Director in Cytogenetics, New York State Department of Health for 7 years from 2000 to 2007. He is a multifaceted personality. He served as a cytogeneticist, Institute of Laboratory Sciences, Poway Medical Center and Hospital Limited, Coimbatore, Senior Scientist, Research and Development, Cancer Genetics, Cambridge, USA, Director, Research and Development Director, Genomics Research and Services, Cancer Genetics, Milford, USA. Group Leader in Cancer Biology, Genome Institute of Singapore, Adjunct Assistant Professor, School of Biological Sciences, Nanyang Technological University, Singapore, Consultant Molecular Genetics, Genome Institute of Singapore, Associate Professor of Pathology, Michigan Center for Translational Pathology, Department of Pathology, University of Michigan Medical School, and Visiting Professor, Department of Food and Nutrition Sciences, King Saud University, Saudi Arabia, Associate Scientist, Vaticuti Urology Institute, Department of Urology, Henry Ford Health System, and at present Associate Professor, Michigan Center for Translational Pathology, Department of Pathology, University of Michigan Medical School, University of Michigan. He tapped the financial grants from various funding agencies and completed 31 research projects, and at present, Health Disparity Research Award Project, comprehensive molecular profiling of prostate cancer in African American population, unraveling molecular heterogeneity is going on. He has received a number of honors and awards. He has been serving as the associate member of American Society of Human Genetics and American Association for Cancer Research, an active member of Society for Basic Urological Research and American Association for the Advancement of Science. His rich experience in research helped him to serve as the reviewer in 17 journals and member of editorial board in 5 journals. Sir is actively involved in teaching as mentor for graduate, undergraduate, and high school students, and mentor for research assistants, research associates, and research technicians. He is acting as the PhD dissertation committee member of Wayne State University School of Medicine, USA, University of Madras, Chennai, Anna University of Technology, Tiruchirappalli, Bharathidasan Institute of Technology, Bharathiar University, Coimbatore, University of Miami, USA. The Tamil Nadu Dr. MGR Medical University, Tamil Nadu, National University of Singapore, and Sri Ramchandra Institute of Higher Education and Research. In his research life, he has been associated with a number of institutional committees at national and international level. In social realm, he actively acting as director, Traver Lakes Homeowners Community Maintenance Association. He has published numerous research papers and shared his research findings in various platforms at national and international level, and acted as resource person in various seminars and webinars. By his non-stop effort, he has received 5 patents for methods of analyzing chromosomal translocations using fluorescence in situ hybridization, RAF gene fusions, and RNA chimeras in human leukemia and lymphoma, and submitted 9 applications for patent. We are blessed to have you in our midst to share your rich experience. It's my pleasure to extend a cheerful welcome to you, sir. I wholeheartedly welcome you, sir. Our joy bounds no limit when we welcome our secretary, Sister Dr. B.J. Queensley Jayanti, Mother Superior. Sister is a person of hard work and always duty conscious. Her initiatives are all encompassing and targeted to achieve the objectives and goals of the institution. I extend a cordial welcome to you, sister. I would like to welcome Sister Dr. S. Jaisarani, the principal, who is unassuming but quick to implement any education and social scheme. I wholeheartedly welcome you, sister. I welcome all the faculty members, research scholars, and students from other institutions and JAC who have accepted our invitation to acquire knowledge and experience from this webinar. A hearty welcome to you all. I wish you all a fruitful experience during this webinar. Welcome, one and all. Thank you.
Thank you, ma'am, for your warm welcome address. Next, felicitation by Dr. Sister S. Jaisarani, Principal, Jaira Jannabakyam College, Periyakulam. I request Sister to felicitate. I request all the participants to be on the video mode for 10 minutes.
Am I audible? Am I audible? Am I
Yes,
audible?
sister.
Yes.
Yes, sister.
Yes, sister, you're audible.
Good morning. Knowledge. But wisdom is organized life. Respected dignitaries of this webinar, Professor Dr. Nallasivam Palanichami, Scientist, Urology Institute, United States of America. Dr. J.S. Yogesh Kumar, Scientist, Zoological Survey of India, Ministry of Environment, Forest and Climate Change, West Bengal. Dr. K. Prempar, Professor and Head, Department of Biomedical Sciences, Bharatidasan University, Tiruchirappalli. Dr. Mohan Raj, Professor and Head, Department of Ocean Studies and Marine Biology, Pondicherry University, Andaman. Dr. Sister P.J. Queensley Jayanti, Secretary of our college and Mother Superior. Dr. Anita Kannagi, Head and Associate Professor of Zoology of our college. Dr. V. Shanti, Convener of this webinar. Lieutenant Dr. L. Merlin Diana, Organizing Secretary of the webinar. Dr. Catherine B. Alexander, Controller of Examination, Associate Professor. Other faculty members of the Department of Zoology, faculty members and students from other institutions. A very warm welcome to you all. Am I audible now? Am I audible?
Yes, sister. Yes, sister.
Every day we see the world constantly changing. One of the areas continuously experiencing breakthroughs are the life sciences. Life science is an enormous field of study that examines every living thing on earth. It explores every single area of our world, the oceans, the earth, the skies, the deserts, the forests, the mountains, and so on. Life science research plays an important role in the research and development of multidisciplinary areas related to living creatures. The combination of multidisciplinary research in the plants, animals, microorganisms, and other interactions with molecular biology. Research has broadened the horizons of life sciences. The life sciences have always been an area dedicated to the protection and improvement of human, animal, and natural life. Now, modern age life science research is more inclined towards interdisciplinary study. A new trend in life science incorporates biological research involving emergence of diverse disciplines such as microbiology, biotechnology, molecular biology, biochemistry, ecology, agricultural, bioremediation, human diseases, and overall. Several biotechnological approaches towards the studies ranging from green photosynthesizing plants to human pathogens. Please, all the members, please mute your mics, please. Please. Life science discoveries are helpful in improving the quality and standard of life and have applications in health, agriculture, medicine, and food science industries. Life sciences research and development has opened up new challenges and opportunities in the related areas like bioinformatics, microbiology, biochemistry, biomedicine, cell biology, etc. Hence, it is the need of the hour to know the recent trends and the emerging developments in the field of life sciences. At this juncture, I congratulate Dr. Anita Kannagi, Dr. V. Shanti, and Lieutenant Dr. Merlin Diana, organizers for this webinar, and other faculty members of the Department of Zoology for their strenuous efforts in planning and conducting this Golden Jubilee Year webinar in a successful manner. Dear participants, I hope this webinar will throw light on emerging areas in the field of life science, and the lecture of the resource persons will kindle your interest in recent research. Let your participation be in a fruitful manner and enrich your knowledge in the topic of the webinar. Congratulations to all of you. Thank you. May God bless you, dears. All the best.
Thank you, sister, for your energetic and enthusiastic felicitation address. Next felicitation by Dr. Sister Quincy Jayanti, Mother Superior and Secretary of Jaira Jannabakyam College, Periyakulam. Sister, I request you to felicitate.
Warm greetings and salutations to you all. It is with great pleasure that I wholeheartedly welcome our resource people, Dr. Nallasivam Palaniswamy, Associate Professor and Scientist of Urology Institute, USA, and Professor Dr. Brain Kumar, Head and Associate Professor of Department of Biomedical Science, Bharatdas University, Trichy. Webinars have indeed become active resources of knowledge dissemination. On this day, I deem it my pleasure to welcome all the participants who are eagerly awaiting to listen to the lectures of the dignified resource people we have amidst us, courtesy of this webinar. This precious time we are investing in here will certainly not go in vain. Wise investment of time can be compared to wise, informed investments in a bank. Though we might not now know when later in the day, they will repay us heftily for sure. Moving forward, I take this opportunity to congratulate and appreciate Dr. Mrs. V. Shanti, Associate Professor of Zoology, the convener, Dr. Mrs. Anita Kannagi, HOD and Associate Professor of Zoology, and Lieutenant Merlin Diana, Assistant Professor, the organizing secretaries, and all the faculty members of Zoology for putting their valuable efforts to organize this webinar for us. Life sciences research plays an important role in the research and development for appliances in the multidisciplinary areas related to living creatures. It generates new knowledge and understanding that provides the foundation for applied research and development. Research provides reliable information in biology for application in the life. More in-depth knowledge through research could be seen as a way to encourage innovative activities. Life sciences efforts go beyond molecular basic science. Improvement also advances behavioral sciences and ecology. The greatest recent advances have been in ecosystem, biodiversity, biotechnology, proteomic and genomic studies relating to metagenomics and healthcare. The combination of multidisciplinary research in plants, animals, microorganisms, and their interactions with molecular biology, genetic engineering approaches, and advances in cell biology research has broadened life sciences. I hope the lectures will have a profound effect on you all. So, dear students and dear participants, please do make prudent use of this opportunity at your hand and listen ears with rapt attention to these eminent dignitaries. May the good Lord be with you and anoint you with the oil of wisdom and intelligence. Wish you all the very best today. Wish you a happy, blessed, prosperous, and new year. Thank you.
Thank you, sister, for your dynamic and divine felicitation address. Now, I invite Professor Dr. Nallasivam Palaniswamy, Associate Professor, Scientist, Urology Institute, Medical Group Urology, Prostate Cancer Research, Detroit, Once again, may I request Dr. Professor Nalasiva Palanichami to give the keynote address.
Let me thank our principal and our Mother Superior and Secretary for providing a platform to conduct this international webinar. We are very honored to invite Dr. Nallasivam Palanisamy, scientist, Neurology Institute, for accepting our invitation in his busy schedule to deliver the keynote in this international webinar. Thank you, sir, for your concern. Please share your screen.
Please, all the members, you meet. All the members, mute your mics, please. All the members, still we are hearing noisy.
Dear participants.
Hello, someone is talking all the time. It is really disturbing.
Dear participants, please kindly mute your audio.
Yes, now okay. Sir, continue.
All right. Hi, everyone. Are you able to see the presentation now?
Yes, sir. Yes,
sir.
Okay.
Your screen is visible, sir.
Okay, great. Thank you. So, wish you all a happy new year. Thank you very much. I would like to thank the organizers for giving me this opportunity to share my work. So first, what I'm going to do, we are going to talk in general. I'm not going to actually overwhelm you with any kind of complex experimental data or anything, since most of you are involved in other biological research and some of you may be involved in cancer research. So just to benefit both students and faculty, I put this presentation in more like in a general kind of theme, so that you will be able to follow easily, because this will mostly this presentation will introduce you some new concepts, and also it will give an overview of the recent developments in terms of actually biomarker discovery in cancer research. I will give you the most updated information so that you can put this information in perspective, so that you can actually think through, and you'll be able to kind of impart this knowledge to your students and other people. So the first, I will present the outline of this talk. So in this talk, what I'm going to do, I'm going to give you a general overview about the cancer incidence worldwide, what is happening, because as you all know, I think the cancer is the major killer disease in the entire human population. I think besides other infectious disease and other things like heart disease and other things, I think cancer is a global issue. So I will present you with a general overview about what is going on all over the world, not only in the US, and also in the whole world. And I will briefly discuss about the major causes for cancer. And then I will introduce the concept of the discovery of the first cancer biomarker. Okay, so as I go through, I will describe each one, what is a biomarker and how it is being used. And now we are in the kind of the best time of research, as the field has actually evolved tremendously, and that we are at a point that we will be able to treat the cancer patient in a personalized or individualized manner. So although cancer is a general kind of term and every individual is unique, and I will talk more in detail about how we can actually, we are doing what is called the personalized medicine using the biomarkers, and how these biomarkers are actually used to develop some therapy, which are actually specifically targeted. So for example, like a patient and identified with a particular biomarker, we can develop some drugs that is specifically targeted for the biomarker so that the patient will get the maximum benefit rather than treating them with the general treatment. And I will discuss about the current challenges in the treatment of cancer, particularly by using the biomarkers. We have a great deal of understanding by looking at the inter and the intra-tumor heterogeneity. I think that is the major challenge why we are not able to kind of treat cancer or actually cure cancer. So as you know, some of your friends or family relatives or somebody affected with cancer, initially they respond to the treatment, but after a while, after 6 months or a year, the cancer comes back, right? Then we are not able to do anything with that. So that problem is mainly because of this tumor heterogeneity, which I will talk in more detail. And also I will touch upon how the biomarkers are used in the early detection of cancer. I think that is the greatest challenge now, because when the cancer is advanced, it is hard to manage. So the earliest, when you detect the cancer, it will be easy to kind of manage. So then finally, I will close this talk by showing you what are the new technologies that are being developed targeting the biomarkers for cancer treatment. Okay. I would like to know how much time I have for the presentation. Is it like 45 minutes or like 60 minutes?
45 to 50 minutes, sir. 45 to
Okay.
50 minutes you can ask.
Oh, okay, I will try to be on time. Thank you.
You can take your own time, sir. That is more, even 1 o'clock, 1 hour, if you want, you can, because your topic is such a nice topic now.
If you allow me, I can go for 2 hours, but I don't want to.
I know the time at 11:30, no, sir. So that also we have to
Okay.
see.
Okay, thank you very much. Yeah. Okay. So to begin with, so this slide, it shows you the worldwide incidence of different type of cancer. So as you can see, the breast cancer is the most common cancer almost all over the world. Okay. That is mostly in women. Then in the men, the prostate cancer is the most common cancer across the different population in the world. And the next comes the lung. And all other cancers are with a less kind of prevalence when compared to I think the breast, prostate, and the lung. These are the three major cancer, that kills more than I would say like 8 to 10 million people every year. So I will talk in more in detail about the numbers, how many cases that you are seeing. So this is the more recent data. So this is actually 2020 data. If anyone want to learn more about this, there is a website called GloboCan Cancer. It is at the bottom, actually left bottom of this slide. You will see the link. It's called GloboCan 2020. You can go to the Google search. You will find the website. You will find even more detailed information. So here is the numbers. So when I say the breast cancer is the most common cancer, as we can see, overall, that is adding all cancer types, there are about 19 million people are affected with cancer all over the world every year. This is not like the total incidence. Every year, we see about 20 million people are affected with all different type of cancer. So like I said, the breast cancer is affecting more than about 2.2 million people. Lung cancer is also about 2.2 million. Then prostate is about 1.5 million people. And the rest of them are with the less numbers. And so given this large incidence of this cancer, so obviously there will be more death as well. So almost like half of these people, affected with different type of cancer, they die because of cancer. So it is not with any other kind of comorbidities. So they may have heart disease or other infectious disease. That is not accounted here. So this is the number of deaths just because of this cancer. Okay, so it's about half of the people. So 19 million total number of cases, about 10 million people are dying because of the cancer. So this is the numbers taken all over the world. So in this map, so So this is to show what are the different countries, what is the prevalence of the cancer incidence around the globe. As you can see, the darker blue is the one showing the countries that shows the very high incidence of cancer. So as you can see clearly that the so-called the developed countries, okay, the North America, the Europe, and then the Russia and China, and then the Australia. So these are the so-called developed countries. Those are the countries have a higher number of cancer. So just keep your eye on the India as well. I'm going to go through several maps. As you can see, in India, we don't have that many number of cases as of now, but there is already I think I heard from many people that there is a significant increase in the total number of cancer cases in India as well, but it is not as alarming as in the other developed countries. Okay, as you can see, the other the so-called underdevelopment or developing countries like Africa. Then some of the Asian countries, the cancer incidence is very low. So here, so obviously when there is a more number of cases, so in this slide, we are looking at the cancer death in which countries, although the developed countries have more number of cancer cases, but you don't see that many number of deaths, because that is mainly because of the advancement in the medical field, the treatment facilities, availability of health insurance, and also the affordability of the patients, who can actually get treated. So you don't see, although the cancer incidence is high in the developed countries, but the number of deaths are significantly low compared to some of the African countries and the China and the other countries. Okay, there is more number of deaths in the other part of the world, but again, just keep an eye on India. Is not that alarming as well. So here in this map, I'm showing what is the most prevalent type of cancer in men and all over the world. Okay, so in this map, all the greens that you are seeing is the prostate cancer. So in men, like we discussed before, the prostate cancer is the most prevalent cancer across the different countries, mostly in Africa, South America, and the North America and the European countries. But in India, as you can see, it is mostly the oral cancer is more prevalent, but not prostate cancer. Okay, since because of our lifestyle, a lot of people are chewing the betel nut and the pond pita or whatever it is. I think that is the main cause that we are seeing more number of oral cancer, which is much higher in India, but whereas the other countries, the prostate cancer is much more in the other countries. So this is the number of cancers in men. So next, in women. So when you look at the cancer incidence, women across the globe, again, obviously the breast cancer is the leading cancer almost in all the countries. And in African countries, the cervical cancer, I think that is mostly due to hygienic conditions, poor living conditions that is associated with the actually cervical cancer, because the HPV, the virus infection, is more common for causing the cervical cancer. I think in some of the African nations, the cervical cancer is more, but in the other countries, mostly the breast cancer is the most common cancer, including India. It's not only, it's almost the entire globe, the breast cancer is the most common cancer among women. So these are all, I think I'm showing this information. I think you can put in perspective that, you can see what are the things that is causing this cancer, how you can avoid all those things, So the next, here I want to show you the number of deaths, because I showed you the incidence of cancer in males and females. Now we want to see the death. So here, the most common cause for death among men is mostly lung cancer, and next comes the prostate. So the lung cancer is more common almost in North America, South America, and the European countries and others. In India, the lung cancer is not that common. Again, the esophageal or the oral cancer is again the major cause of death in men in India. But whereas the prostate cancer is more common in Africa and the rest of the world, the lung cancer is the major cause of death, because this may be due to heavy smoking. Is the smoking, marijuana, and other. I think the major problem is that tobacco use is very high in those countries. I think that is the reason the lung cancer is the leading cause of death in men. Then what is the cause of death in women? As you can see, it is little kind of different. In most of the countries, all the pink is the breast cancer. That comes in India as well. The breast cancer is the major cause of death among women in different parts of the world. And like I said before, the cervical cancer is more common in African countries, which I told you about the hygienic conditions and the health problems. The cervical cancer is the major cause in African nations, but the lung cancer is mostly in because I think women in the African American countries, they are also heavy smokers, when compared to the Asian and other countries. So I think that is probably the reason why the lung cancer is also a major cause of death among women in the so-called developed countries. Okay. I think that is enough for the global incidence. I hope that will give you an idea, what's going on in terms of actually overall cancer incidence across the world. So now, I think based on the rate of increase in the number of cases, it is estimated that in about 20 years, the number of cancer cases is almost going to be another like 10 million cases more. It's going to be in the year 2040. We are going to see about 30 million people are going to be affected with cancer. I think that is the kind of very conservative estimate that they are projecting. Okay, so so although we are actually heavy investment is going on for cancer research and other thing, despite the cancer incidence is keep growing. I think that will, I hope that will create an awareness among the people to see how they can actually control the incident. So talking about the global incidence in male, female, different type of cancer. In this slide, I will show you what are the kind of the major causes of cancer. Okay, because it is not just one factor that is actually causing cancer. There are so many. Okay, so I think this will actually give you an idea how you can actually limit your exposure to all these risk factors so that you can avoid actually getting cancer. First, of course, most of the old age is a major factor. Most of the cancer occurs in the older people because the cancer is not like an infectious disease. Cancer, it takes about 10 or 20 years in the making. The early stages, the cancers are very silent. Only in the later stage of your life, when it's advanced, it started showing up symptoms. Then only you get diagnosed. So the older people are getting more cancer. That is a common. Then alcohol use, that is another major contributing factor. And cancer causing substances. I think mostly, now we are talking about a lot of these kind of Western food, they are putting a lot of preservatives, additives, and other things, those are all a lot of carcinogens being included in the food. And moreover, we are using a lot of pesticides, when you're raising crops, whether it is fruits or vegetables or whatever it is, they are heavily kind of, there is a heavy use of different kind of pesticides, and these pesticides are carried over through the food, and that is getting accumulated in your system. And most of those pesticides are carcinogens, that's the reason some of the cancer causing substances are getting into the food. So we have to be careful what we are eating. And inflammation, diet, of course, then different hormones, immunosuppression, infectious agents, obesity, and all those problems. So I think the workplace, we have to be very careful. We have to provide a very healthy environment in terms of air and the water and all the surroundings, even the workplaces and most of the industries and other things, at least in the Western world, there is a lot of strict control measures are being implemented, but I don't know, these kind of rules are strictly followed in the developed countries, mostly in the Asian and the Indian subcontinent. So the rules are not strictly followed. As you can see, a lot of rivers and the water bodies are getting polluted. I think that is the major cause for cancer. Next, industrial pollution. As you can see, like the developed countries, they already shifted most of the kind of the major industries are shifted to the Asian countries. So the most of the productions are done in the Asian countries and exposed to developed countries because they knew that it is the major issue, the industrial pollution. They emit a lot of toxic gases and fumes and other things, that affects the major tragedy that happened in Bhopal. As you know, the company called Union Carbide, they released this toxic gas that killed so many people. So like that, when people are living around the industry, they should be very careful. They should take some kind of precautionary measures to avoid any accidental exposures. And also the Chernobyl disaster, in Russia, that also caused a major problem. So these are all the major problems. And the sunlight, especially, this is not a major problem in the Asian countries because our body, it synthesizes good amount of melanin that gives protection. But in the Western world, especially the Caucasian people, their skin is white. The incidence of skin cancer called melanoma is very high in the Western world. So the sun exposure to sunlight is a major problem. So that's why they sell a lot of these actually what is called creams with the SPF, what is called sun protecting factor. They add it into the cream. They apply to the skin before they go to the sun, like that. So that is another major problem. Then, like I said before, tobacco. Please avoid smoking or any form of tobacco is not good. Okay. Avoid that. Then alcohol and tobacco, again, the problem. Then diet, again. So you have to be careful on what you're eating, So like red meat or any food with a high fat content and all those things, they're not good. So I think just by eating the diet over a period of time, because this is not one time or it's not going to affect, but when you actually follow this kind of careless diet over a period of time, there is definitely there is a potential risk. The next, some of the cancers are caused by viruses, like the cervical cancer we talked about. Those cancers are caused by the HPV virus. The herpes simplex virus is the major cause for cervical cancer. So I think following strict hygienic conditions is important to avoid cancers due to these viruses. I think mainly to avoid all these things, I think that's why, especially in Western countries, they regularly do the pap smear every year. All the women go through the pap smears in a kind of screening, make sure that they're not getting any cancer. And the next is, like the diet, just eat more greens and fruits and vegetables. And I think that is the best way to have a healthy diet. All right. So I think enough for preaching on the different causes. So coming to the point now, what is cancer? I think cancer is a broad term. It is it just encompasses everything. It is simply a normal cell is transformed into an abnormal or a more continuously proliferating cell. Once a normal cell is transformed into cancer, it never stops growing. It will keep on kind of growing everything. So basically, cancer is a broad term, but it is a group of all genetic diseases that result from changes in the genome. So I put it in simple terms, cancer is a disease of the genome, meaning that there has to be some change in your DNA. Okay, it could be at the chromosome level or at the gene level. There has to be some change in the DNA that causes cancer. So without that, you are not going to get cancer. So the entire genome is completely affected. I will talk more in detail about what are the specific changes that will cause cancer as well. So uncontrolled proliferation is the major reason for the cancer. So this is a broad kind of definition I'm putting here. So when you look at, as I said, the cancer is a disease of the genome. When I say genome means it's not just the DNA alone. The DNAs are packaged in the chromosomes. So there are so many changes or so many cancers that are associated with kind of chromosomal changes. So like some of the chromosome changes are called structural chromosome changes. As you know, humans, we have 46 chromosomes. So these 46 chromosomes, they are not kind of present in a proper order, okay? So there will be some breakage and joining, like that is called translocations, so which I will show you in a couple of slides after. And some regions of the genome will be deleted, and some regions of the genome will be duplicated. Then some regions of the genome from one chromosome, it will break and get inserted into a different location in the genome. Like that, you know, there are so many complex changes that goes in the cancer cell. You will see it only in the cancer cell, but not in the adjacent benign cell. So these type of cancers are more like leukemia, lymphoma, multiple myeloma, and some of the solid tumors. They carry all these type of chromosomal changes. And some cancers are caused by numerical chromosome changes, meaning the total number of the chromosome changes in those cells. So when you look at those cancer cells, you will not see 46 chromosomes. It's definitely more than 46, you know. So if you look at some of the cell lines, or if you put any cancer cells in culture, we end up seeing that minimum like 60 or 80 or sometimes 100 chromosomes in each and every cell. Such a complex. I will show you some examples later. And besides the known chromosomal abnormalities, there are specific mutations, you know, just a single gene or single base pair change also responsible for causing cancer. So here I want to show you the first chromosome abnormality in the history of the cancer research. So the first chromosome abnormality that was known to be associated with cancer. So rather I would say that this is the first biomarker, okay, that is identified to cause cancer. So these are the two leading scientists. It's called Peter Nowell and Hungerford from the University of Pennsylvania in the year 1960. They identified a chromosome 22, which is always consistently smaller in the patients who are affected with a type of leukemia. It's called a blood cancer. It's called chronic myeloid leukemia. That was in 1960. They first identified that consistently one of the chromosome 22 is always smaller. They thought that, so because there is another normal chromosome, which is much larger, but there is always, they see a small chromosome. They thought that chromosome is, a portion of that chromosome is deleted. This was the observation in 1960. Then in 1973, the Dr. Janet Rowley from University of Chicago, she did a more detailed chromosome analysis. She confirmed that the chromosome that appears smaller, that was identified by Peter Nowell and Hungerford, it was not actually deleted, but the portion that is missing in the small chromosome, in fact, it got translocated to a chromosome 9. Okay, so this is the normal chromosome 9. Are you able to see my cursor? Okay. Sound.
22, your cursor is now 22, no?
Okay. So the portion that is missing from chromosome 22 is actually translocated to chromosome 9. So there is no loss of any DNA or anything. In fact, the portion is actually misplaced, you know, instead of chromosome 22, it went to chromosome 9. So here I will show you, so this is the normal human karyotype where you have the 46 chromosomes, the X and Y chromosomes. This is a perfect normal karyotype, but when you look at the karyotype of a patient with chronic myeloid leukemia, essentially all other chromosomes are perfectly normal except this long chromosome, chromosome 9, and there is a chromosome 22. The portion that is missing in 22 is translocated into chromosome 9. So this is the only abnormality that is happening in the cells of this is particularly in the white blood cells. Okay, this all these changes are happening in the bone marrow, what we call hematopoietic stem cells. These cells, have this mutation, and all the white blood cells that is produced from the hematopoietic stem cell carry this translocation, and they produce an abnormal protein that is responsible for cancer. So any patient comes with the leukemia, they always look for this particular chromosome translocation. So this is the first biomarker that is identified in cancer. Okay, that is highlighted. So this is to show what exactly happening between chromosome 9 and 22. So there is a gene, or the ABL is located on chromosome 9 in this location, the yellow. Then the BCR is called breakpoint cluster region gene. This is on chromosome number 22. So basically there is a break in this chromosome 9 and also on 22, and they exchange bases. The portion of the 22 goes to chromosome 9, and the portion of the 9 will go to chromosome 22. So this is called a balanced reciprocal translocation. So the point where the breakage in the chromosome occurs, it affects the gene. It goes through the gene. Okay, here on chromosome 9, ABL gene is affected. On 22, BCR gene is affected, and a portion, because it happens in the middle of each gene, and the segment of these genes are exchanging the places. It creates what we call as a fusion gene. Okay, this you will see only in the cancer cell, but not in the normal cell. So now, using this, I'll go through this history here. As you can see, first it was discovered with this small chromosome. Then we know what is going on between 9 and 22. They cloned the gene, the BCR able. Now we have a drug. It's called STI-571 or Gleevec. This is the drug because this gene, the ABL, is an enzyme. Okay, so by using this drug, they can actually inhibit the tyrosine kinase activity of this gene, or rather it is the protein. Okay, the tyrosine kinase protein will be inhibited by using this drug, because only this ABL protein goes to abnormal level in the cancer cells. So by using this drug, you can actually block the activity of this enzyme so that the patient will be relieved with the symptom. So this is the very remarkable and important discovery, starting from the identification of the chromosome to the development of the drug. So even the patient, because this drug was actually discovered in 1999, even the first patient who received this drug, she is still alive today. All she is doing is she is taking a 400 milligram pill every day, and almost like 20 years now, she is alive. So here I want to emphasize that you cannot cure the cancer. Okay, but you can actually manage the abnormal level of the ABL by taking this drug, and you can live life. So this is a remarkable kind of discovery that was made by simple observation of the chromosome to the development of drug. So this is a very simple and elegant example. Okay, but so that is the leukemia. That is the cancer in the white blood cell. So now I'm going to show you an example how the cancer cell will look like in the other solid cancer, meaning that the cancer in different organs. The first, so this cell is coming from a liver cancer patient. As you can see, like we have a method, what we call spectral karyotyping, that we can visualize each chromosome, all the 24 chromosomes in 24 different colors. So that you can see whether you are looking at a normal chromosome or an abnormal chromosome. If it is a normal chromosome, the entire chromosome will be represented with one color. But if you see any chromosome with more than one color, that means there is a breakage and joining. In this example, there are multiple breaks here. It's not just one. As you can see, there are many chromosomes that has complex chromosome translocations. And similarly, this is another sample from a breast cancer patient. Just imagine here, there are more than 80 chromosomes in this cancer, and there is almost, there is no normal chromosome here. Almost every chromosome is involved in this breakage and joining, okay? So this is the major challenge, okay? So it's not like the other case like CML where we have only one simple translocation, but here there are so many. So we don't know which one is really responsible for causing cancer, okay? And moreover, if you look at this translocation, it is not the same in all the breast cancer patients, okay? Every patient will have a different type of breakage and joining. That is the major challenge to identify which translocation is actually causing cancer. That is the biggest challenge that we are facing. And moreover, every cell in this patient, in the cancer, they have entirely different chromosome complement. They are not completely identical, you know. So that shows the enormous complexity of the cancer cell. So I will talk more in detail about in the subsequent slides. So here, so basically, identification of these kind of chromosomal aberrations, you know, identifying actually specific chromosome abnormalities, specific cancer is what we call it as a biomarker. Okay, so it has to be one chromosome translocation involving a gene that produces an abnormal protein. So you are looking for a molecule that is specific in a tumor cell, and that can be targeted with the drug and other things. So that's what we call it as a biomarker. So the biomarker can be classified in different categories. So one is like diagnostic. So if you identify, so like the example like chromosome translocation, if you see that particular chromosome translocation, then you can call it as that is the particular type of cancer. It can't be anything else. So that you can use those markers to diagnose this cancer. So any patient go to the clinic, first they take the biopsy, they do all these tests and they confirm what is the type of cancer. Then you can actually go with the, what is called prognostic. So when you have this marker, what are the drugs available and how likely the patient is going to respond to the drug, unless you know which drug targeting which biomarker you are giving. Otherwise, you know, you are shooting in the dark. You know, the patient is not going to get benefited. So like that, there are different type of biomarkers that you can identify. Here I will show you a list of the biomarkers associated. This list is exhaustive. I just to show you a few examples. Like these are the specific kind of markers associated with the different type of cancer. If you guys need this slide, I can share it. We don't have to. But if you like, I can share this slide later. So just listen and follow this. So here, for each cancer, these are the most commonly identified biomarkers. So any patient goes to the clinic, immediately they perform this biomarker test, make sure that, you know, these patients have this type of marker. Then you can say that, like people with ovarian cancer, so simply they what is called the CA125 marker is an antibody test. They will so when you see this level will be very high, it will be like 600 or 800 units like that if the cancer is advanced. So like that, each marker is diagnostic for a particular type of cancer. So now I think nowadays any cancer patient go to the clinic, nobody is getting treated without doing a kind of some proper test as well. So by using these markers, what you do that when you have the biomarker, so here I'm showing an example on the prostate cancer that we are working on. So first, when the patient comes to the clinic, they take the initial biopsy and they run through all these different tests, because we know these are the different type of driver mutations that are identified in prostate cancer. So when I say that these many markers, you're not going to find all these markers in one patient, okay? So when you find, say for example, P10 abnormality, you will find only P10 abnormality in that patient. You won't find other changes. So basically, by identifying different biomarkers in a cancer type, you can classify the patients into distinct molecular subset, or you can actually stratify the patients, you know, the group. So if you have 100 patients, like 10% of the people have one mutation, other 5% have another mutation like that. So that, but you have to screen for all the mutation from the initial biopsy. So which type of mutation this patient has. Then there are actually specific drugs developed for each marker. Then the patient will be treated with that particular drug based on what is the aberration that is there. But of course, all these aberrations that are present in only a small subset of patients. Okay, still for prostate cancer, we identified only about 50% of the patients are identified with marker. For the remaining 50%, we don't know because those patients do not carry any of these markers. So that's where we are focusing our attention to discover more biomarkers, so that otherwise, if we don't have any biomarker, these patients will receive the more generalized chemotherapy or radiation therapy because they just want to control the proliferation. But if they carry this biomarker, you can give the specific drug, the cells that is carrying this particular mutation will get eliminated so that the patient will get the benefit immediately. So even though you have these biomarkers and the drugs, everything, sometimes even if you give the drug, some of the patients don't respond also. Okay, even if they carry the mutation. It is because of that complex other chromosomal changes. Some of the patient, or initially they may respond and the disease may come back. If they come back with the disease, then we have to do what is called a repeat biopsy. You have to take the biopsy again, then do all the kind of the molecular screening again to see if there are any new mutations. Then based on that, you can actually give some new drugs. Or if you find entirely new mutations, the drug may not be available. In that situation, the patient has to go through the standard chemotherapy or radiotherapy. So these are the kind of the use for the biomarkers. Okay, so that's why the extensive research is being conducted for each and every patient. We are doing extensive kind of screening by doing the whole genome sequencing and other type of analysis. We are looking for any specific mutation. So importantly, if you find a biomarker, it means if you find one mutation in one patient, that is not important. You have to find this mutation across several patients. Then only, those markers will become kind of what I would say a bona fide biomarker. It has to be, in other words, the mutation has to be recurrent, meaning that it should be present in more than say like 1% or 2% of the patients. Then only it has some significance. Otherwise, it is more like a private mutation that is present only in that patient, and you may not find the same mutation in another patient. Okay, so those are all the other challenges that we are facing. So once you identify all these markers, so that's what boils down to what we are doing right now is called personalized medicine or the individualized medicine. Okay, so basically each patient come with a different type of kind of mutation or whatever it is. Then we have to do the extensive screening by these are different methods like either whole genome sequencing or a PCR method or fluorescent in situ hybridization or immunohistochemistry for antibody. Depending upon the mutation, we perform all these different type of tests, identify the patient with a particular type of mutation, then we select them for kind of the targeted therapy. So this is the current practice right now. It is being going on. So anyone is interested, you can go to the Google search. You will identify what is called the NCCN guideline, the National Cancer Care Network. So that gives the complete kind of biomarker for each cancer and what are the available drugs available for that. So I think any patient nowadays go to any clinic. I think even in India they are doing it. They run through, since for each cancer type there are several biomarkers are identified. So they go through extensive screening and identify the right patient with the right treatment. So when we talk about biomarker, it is not, like I said, it is not always only DNA. It could be the kind of changes in the DNA, like gene mutation, other thing, or sometimes in the RNA as well. So some of the non-coding RNA that recently we found out that is also overexpressed or abnormal or it is deleted. Those are all cancer biomarkers. Sometimes, the proteins, the abnormal proteins, like I told you, some of the fusion gene that causes some abnormal protein. And recently, from our group, we identified some of the metabolites. Okay, so I'm sure in the class you are teaching about the Krebs cycle and all those, what is called the TCS cycle and all those things. During the several pathways, and it gets that pathway get affected, then the metabolite that actually started accumulating in the cancer cells, not in the kind of normal cell, but in the cancer cell, several metabolic pathways are disturbed, so that the metabolites get accumulated and they become they can be used as a kind of as a biomarker for diagnostics. So it all depends upon which cancer, which type of cancer and what are the changes going on. It could be DNA, RNA, protein, or metabolite. Okay, so you can identify a biomarker in any of these categories depending on the cancer that you're looking at. And recently, even the abnormal level of lipids also known to be associated with particular type of cancer. It's called lipidomics. That is a very kind of emerging field. A lot of research is going on. This is being done using the mass spectrometry type of analysis. Okay, here let me show you more specific cancer. What are the kind of biomarkers that we are seeing in one cancer? So, okay, here I'm showing an example of lung cancer. As you can see, we identified several mutations. There are actually 15 different types of mutations are known to be present in the lung cancer patient. So like I said before, you will find only one of these mutations in a single patient. You are not going to find all the 15 mutations in a single patient, okay? So you will find either any one of these mutations. If you find, but initially, once you get the biopsy, they screen for all the mutations to see, because they don't know which one is there. Because based on the kind of biopsy, when the pathologists look at it, they confirm it is cancer, but they cannot tell what type of genetic mutation it is carrying. So that's why we have to do this in-depth molecular analysis. So once you identify that, what type of the mutation is there, and based on that, the specific drug can be used. So here in this slide, it shows when you have this particular mutation, there are actually specific drugs. This is all erlotinib, geftinib, and quercetinib. So these are all the kind of specific drugs that are developed targeting specific mutations. So like if the patient is positive with the EGFR mutation, it's about 17% of the lung cancer carry this mutation. So you can choose any one of these drugs. So immediately the patient will respond. The cancer will melt like a wax, okay, when you give the drug. So like that, for each biopsy sample has to be screened for the specific mutation. Then the doctor can select the specific drug and immediately you will see the benefit like that. So here, this is another one for melanoma, because in America, the actual Western countries, the melanoma or the skin cancer is more common. There are so many drugs. I think only recently they identified this biomarker, what is called the programmed death gene, the PD-1. This gene is known to be overexpressed in melanoma. This is basically affecting the immune system. So what is happening is when this gene is overexpressed, our body doesn't recognize the cancer cell as a foreign body. So our immune system allows these cells to grow in our body just because of the overexpression of this gene. So by targeting a drug to inhibit this PD-1 gene, so once you reduce the level of the programmed death gene, then the immune system recognizes the cancer cell as a foreign, then immediately kills those cancer cells. So actually, one of the American president, Jimmy Carter, recently he was diagnosed with the very advanced stage of melanoma, and he was given this anti-PD-1, this called immunotherapy, and he was completely, kind of actually cured of cancer. He's still alive, but he's cancer-free. So that is the remarkable kind of advancement. That because this was ignored for a long time, because people, although they thought that immune system may play a role in cancer, but recently this was actually a major breakthrough. And I'm sure you all heard in the news that these two individuals are the ones actually who made this remarkable discovery, and they got the Nobel Prize the year 2019, I think, they got this Nobel Prize for developing the immunotherapy. So which is a major breakthrough. So now what is happening is almost all type of cancer samples are actually screened for this PD-1 antibody if it is overexpressed, and they as an option, if the patient exhausted all the other options of treatment, as a last resort, they try the immunotherapy as well. But unfortunately, it is not working for everyone. Okay, it works again. Only a subset of patients are getting benefited with immunotherapy, but still there are many patients, and they are not responding to the immunotherapy as well. But of course, there is a good number of patients are getting saved with this. It's a remarkable kind of discovery. Okay, so with all those things, I told you about the different chromosomal changes, different biomarkers, all those things, then we have targeted therapy, immunotherapy, everything. Then why we are not able to kind of help the patients, completely cure the cancer, all right? So as you know, some of your relatives or friends or, or in the news, you see people, initially they're diagnosed with cancer and they undergo treatment for some time. Then they will be cancer-free maybe for 6 months or a year. Then the cancer is coming back, right? When the cancer comes back, then there is no way you can actually treat them. So this is a major challenge, okay? So despite all these drugs and everything, we are able to extend the very advanced cancer patients, the life maybe a year or two. We cannot keep them completely live longer. So this is the major challenge, more research being going on. So here I will show you a clear example. What is the challenge? Why we are not able to cure cancer. So here I'm showing an example, a patient who is 38-year-old man. He has a cancer called melanoma, that is skin cancer. This cancer is known to have a mutation in the gene called BRAF, which is a kinase. This gene, more than about 50 to 70% of these melanoma patients carry this just one base pair change, okay, one amino acid change that completely alters the kinase activity of the gene. So this is the patient when they got the initial biopsy, and they screened for this mutation because he has the skin cancer all over his body, okay. And recently, about 3-4 years ago, there was a drug targeting this BRAF mutant protein. This is called PLX4032. This drug was developed that will affect only this BRAF mutant protein. So when the drug was developed, this patient was treated with this drug. What happened? Within 15 weeks, this cancer is completely gone. Okay, we were able to completely eliminate the cancer with this drug because we identified this BRAF mutation as a biomarker, and the specific drug was given. All the cancer is gone. But unfortunately, after the treatment, within 23 weeks, okay, within 6 months, the cancer came back in this patient. So wherever initially the cancer was there, then the cancer came back. All over the body, the same locations, the cancer came back. So they have only for 6 months he was cancer-free. Now the cancer came back. What are we going to do now? Okay, so now they have to do the repeat biopsy, and they identified they could not find, although they found several mutations, but unfortunately there is no drug available to target those new mutations. So that is the biggest challenge. Most of the solid cancer, whether it is a cervical cancer or ovarian cancer or breast cancer, lung cancer or prostate cancer or liver cancer or esophageal cancer, these type of cancers, we have a minimal kind of response. Initially the tumor will respond to the treatment, then the cancer is coming back. The patient immediately, after a while, they succumb to death. So this is the major challenge in cancer research. So any youngsters who are listening to this presentation, if you're interested in research in biology, please, just read more and more, and that, you can make some breakthrough, doing some more advanced kind of cutting edge research. So I think the whole research world is doing as much, working so hard with the available technology and the resources, but still, I think when we do more research, we are getting some clues, but those things, it really poses more questions than answers. Okay, even when we apply some advanced technology like next generation sequencing and all those things, we are end up getting more questions than answers. Okay, so that is such a complex disease. It's not like infectious disease or other thing, but cancer is a very complex disease, and it is very difficult to control. Okay, so why, so not to explain why the cancer is coming back. Okay, so I'm trying to answer that question. So showing an example here. So using prostate cancer as an example. So this is not limited to prostate cancer. It is applicable to other cancer as well. So what is happening is, so initially, this is the structure of the prostate. So it takes about 10 to 20 years in the making of cancer. Okay, so there are different tumors that occurs in the different locations within the prostate. As as the tumor progresses, it started accumulating more and more complex aberrations there. Okay, so when you have it, so now within the prostate, we are not dealing with one cancer. Okay, so there are actually more than like 3 or 4 different type of cancer within one patient within a single gland. Okay, so now I will show you some examples that whether when you have multiple tumors, are they genetically identical or different like that. Okay. So here I'm showing an example. So this is the work done in my lab. I'm showing a cross section of the entire prostate. So on the left side, as you can see, all the green circles are the 5 different tumors. Okay, and the different locations within the prostate separated by a normal benign tissue. Okay, so this is one tumor and 5 different tumors. By using the different biomarkers that we identified in prostate cancer, when we screen, what we found was each tumor is different. They are genetically different. One tumor has one biomarker, other tumor has other one, like that. And some tumors, we don't know what is the marker. It is completely negative for all the known biomarkers. Okay, so that's why I told you, all the biomarkers that we identify in prostate cancer, they account only for about 50% of the cases. Other 50% we don't know what is going on. So that is the major challenge that's going on. So like you see, there are 5 different tumors with 5 different markers. Okay, but we don't have drug for all the markers. So even if you have drug for one marker, when you treat the patients with that drug, it will eliminate only the cells that carry that mutation, but other tumors, they will start growing. So this is the major challenge. So even though if you identify a biomarker, that is not present in each and every cell of the cancer patient. Okay, so when you give a drug, it will target only the cells carry that particular mutation. So that will be eliminated with the treatment process, but the other tumors, it will start growing back. Okay, so when the cat is away, the rats play. I think that's a simple example. Okay, so when you remove the major clone or the targetable mutation, then the other small tumors, they started growing. That is the main reason the patients initially they respond to treatment, then they come back with the cancer. Okay, and which is not easy to manage. So here I will show you example what I'm talking about. So this is the entire prostate gland. So there are different regions with I will zoom in this one. We actually stained this entire prostate tissue with the different kind of biomarkers. If I zoom in, you will see it. See here how these cancers are distributed. So the area that is stained with the brown color is one biomarker, is called ERG gene. So the mutation is present only in this area. Right next to that, these are also cancer, but that is positive for another biomarker called SPINK1. And in between, this is a normal tissue here. See how heterogeneous it is? So this is what we call as tumor heterogeneity. It's called inter-tumor heterogeneity. So now if I have a drug, say for example, ERG, so the drug will work only on these cells. It is not going to touch the cells with the SPINK1. Okay, so you can eliminate only these tumors, but the other tumors will come back. So this is very simple kind of way of showing what this is the major challenge, why we are not able to actually cure cancer. Okay, So I hope this will actually put your thoughts in kind of proper perspective. You can ask more questions like that. Now, so far I talked about some of these mutations and biomarkers and all those things. Those are all kind of genes that produce a protein. Okay, that is, a gene can be, after transcription, it goes to mRNA, then it translated into protein. So now what we found was in the human genome, more than 90% of the human genome, we have another class of RNA, what we call non-coding RNAs. Okay, these non-coding RNAs, meaning that they don't code for any protein. These RNAs are transcribed, but they never get translated into protein. So that's why we call them as non-coding RNA. There are about 50,000, more than 50,000 non-coding RNAs in the human genome. So that is these non-coding RNAs, although they are not coding for any protein, but they play an important role in regulating gene expression and other things. Okay, so this is what I was telling you. Why the cancer, is getting more complex and complex. Okay, because until recently, I'm sure most of I think senior professors, you might have actually taught to the students when you're teaching genetics. You may say that there is a term called junk DNA, right? Some of the genome, we don't know what is the function of the DNA. Until recently, people thought those are all junk DNA. But there is no such thing as junk, because our genome is evolved over millions and millions of years. But all these sequences are there for a reason. Okay, so I don't think the genome will keep it as a junk there. So now we have a good understanding that the entire genome, okay, more than 99% of the genome is transcriptionally active. There are more and more non-coding RNAs are produced. And we started learning more and more about these non-coding RNAs. So I think, let me go through quickly. So this is a one class of non-coding RNA, what we call pseudogenes. Okay, the gene and the pseudogene. I don't have time to explain all the details. I think the simplest definition for pseudogene is it was first discovered in 1977 from this frog, Genopus laevis. I think the zoology professors, you can very well recognize. Only from this frog, they first identified a gene which resembles a gene that was functioning a long time ago. So that's why they call it a pseudogene. It has all the components, but we don't know the function. So that's why they call it as a pseudogene. So basically, the simple definition is the pseudogenes are dysfunctional relatives of known genes that have lost their protein coding ability or not expressed at all. Okay, so that is a simple definition. Because the sequence will be the same. The wild type gene and the pseudogene, they have a similar sequence, but they have different function. That's why we call it the pseudogenes. So there are different classes of pseudogenes. I don't have time to go through all those things. But recently in my lab, I identified a particular pseudogene. Okay, so this is called a KLK P1 that is involved in, there's breakage and joining, there is a fusion. So the KLK4 is a protein coding gene, and a portion of the KLKP1, which is a non-coding RNA, but after break, so the first two exons of the KLK4 is fused with the exon 4 and 5 of the KLKP1. The KLKP1 is a non-coding RNA and the KLK4 is a protein coding, okay? So after the fusion, what happened, the portion, all the amino acid sequence, I put it in red here. These amino acids are coming from the non-coding, the KLKP1. So after the fusion, the non-coding RNA is converted into a protein coding RNA, okay? So which is unique. And first time we showed that, which I actually published recently, we know that this KLKP1, it is expressed only in the prostate tissue. There are actually 53 different type of normal tissue. If anyone is interested, go to this portal called GTEx portal. You will find the gene expression across 53 different type of normal human tissue. Okay, so here we show that the KLKP1 is expressed only in the prostate tissue. That shows the association of prostate cancer. I didn't have much time to go through the details. So we cloned this gene, we completely identified the sequence, we have the amino acid sequence and everything, we developed antibodies. And the other interesting thing is we can identify this fusion gene in the urine of prostate cancer patients. Okay, so recently we developed a test to screen this prostate cancer patient. They can give just a urine sample. I can screen this fusion in the urine, then they say that whether they have the prostate cancer or not. Okay, so we can use this as a biomarker for screening prostate cancer, the more non-invasive method. So this is the work I published recently. If anyone is interested, you can read the whole paper there. So the last few slides. So now I think you have a good idea about what are the different biomarkers, what do they do, what are the treatments available, everything. And I showed you that what are the challenges to why we are not able to cure cancer or treat patients in a more effective way. So the best way is when the cancer is in a very advanced stage, it is very difficult to treat or manage this patient. What we call metastatic cancer, like when the cancer actually spreading to different parts of the body, it is very difficult to control. That's why now there is a lot of effort and the research is going on for the early detection. Okay, even though the patients are asymptomatic, suppose if the cancer is running in a family, if more than one individuals are affected with cancer, so I think it is better to get screened earlier. Okay, so just by taking the blood samples, there are good tests that are there, so that when you do the early diagnosis, you can improve the cancer outcome by providing care at the earliest possible, and is therefore an important public health strategy in all settings. So I think a lot of effort, a lot of investment is going on. In our lab also, we are doing a lot of work on that for early detection of cancer. And for doing the early detection, the one method is actually looking at all we need is just the peripheral blood. Okay, so we developed what is called the microfluidic device. So you can run all we need is just 1 cc of blood. You don't need a lot. Just take 1 cc of blood, run through this microfluidic device. You can capture the cancer cells and you can separate the white blood cells as well. I will show you an example here. So this is the microfluidic device where you have all these grooves here. When you run the blood, all the cells will go through this. At the bottom of this groove, there is a hole there. So we adjusted the size of the hole so that only the small cells, like white blood cell, can pass through. But the cancer cell, because of the larger size, they cannot escape through the hole so that they will get captured. Okay, so this is a clear example I'm showing that because then once you capture the cells, you can stain with some antibody, which is specific for epithelial cells and white blood cells. The CD45 is a marker for white blood cells that will stain in red. And the cancer cell, this epithelial cell marker CD45 or the cytokeratin. The CD45 is negative because this is specific for a white blood cell. The cytokeratin is positive for epithelial cell, and this is the cancer cell. So you can capture this cancer cell just by taking the circulating blood. Even if you identify one cell like that, that is indicative of cancer. Then we can actually take this particular cell, we can isolate RNA or DNA from this, and we can do some molecular analysis, look for any of this biomarker that we identified. Okay, so this is the way to do what is called early detection of cancer. So this work is going on. So this is the simple device, that we used in our lab. I will show you additional examples here. Okay, so this is a panel. You can stain the cells with the DAPI, just a nuclear stain, the cytokeratin and white blood cells like that. And another method that is recently developed, what we call cell-free DNA. What is cell-free DNA means? The cell-free means there is no cell membrane or anything like that. These are all DNA fragments, okay? Just fragmented DNA that are actually released from dead cells. It could be cancer cell or normal cells. They'll be floating around in the circulating blood, okay? So you can actually just collect the blood and you can isolate the cell-free DNA, and we can do several tests like sequencing or other PCR or any other method, look for the known biomarker mutations that we discussed before. If those mutations are there, so there is an indication that these patients may have cancer so that you can actually capture or do the early diagnosis of cancer using this DNA. And recently there is a publication. It is not completely out yet. It's only the abstract that was presented on June 2020. There is a test. They screened about 10,000 individuals, particularly women, and they were able to identify cancer. Because these individuals, there is no symptoms or any cancer or anything like that, but they are the so-called apparently healthy individuals. They volunteered to give blood, and those blood samples were screened, and about 10% of the patients are captured with cancer. So once they identify these kind of cancer cells in the blood, then immediately the patients will go for more advanced testing, like a CT scan or a mammogram or whatever it is, then you will be able to diagnose when they get cancer. So then you can actually either do surgery or a chemotherapy or radiation at the early stage before the cancer spread to other organs. You can stop it so that the individual will be saved from the what is called the metastatic disease. Okay, so this is the area that is advancing. I think this is the last slide I want to show you. This is the technology. I'm sure you are all teaching in your course as well. It's called a CRISPR technology, clustered regularly. This is the technology for which the scientists got Nobel Prize in 2020. So basically, I'm sure I don't have to explain the details. You can read. The CRISPR is the bacterial immune system. Okay, so they can use this to edit any particular gene in the human cells. Okay, suppose the cancer cells carrying a particular mutation, and you can actually use this CRISPR system and to correct this mutant gene, and using that you will be able to kind of correct the defect in the cancer cells and try to actually kill those cancer cells like that. So this is the more recent kind of development, because only just this year this technology got Nobel Prize and all those things. Very extensively being used in the kind of research lab, but it's not kind of fully developed as a diagnostic test yet. But eventually, I think this is going to make a huge impact both in the diagnosis and the treatment as well. Okay. Because in order to use this technology, you need to know the specific mutation or the biomarker, okay, which gene or which biomarker you want to target. Unless you know, you cannot use this technology. So this is more targeted. Once you know the defect, then you can actually send this technology and this approach to correct the defect in the cancer cells. So I think the last slide, I think we are in the very exciting time of cancer research. Because of the advanced technology and other things, we have a very good understanding about the complexity of the cancer, but still we have a long way to go to find a cure for cancer. But at least we are at the stage that with the extensive molecular screening, we are able to classify the cancer into distinct molecular subtypes, okay, based on the specific markers, so that using those markers, we can develop some drugs. I think the challenge is, even though if you identify a mutation or a particular protein, it is challenging to develop a drug, because when you develop a drug, the issue will be, it has to be specific. It should affect only that protein. But unfortunately, the side effect or the off-target effect of the drug is a major problem. Okay, when you treat, the patient will have a lot of side effects, complexities, allergies, and other side effects. So the patient may not be able to tolerate the treatment. Okay, so like recently, that's what happened with the COVID vaccine also. They have to do extensive tests about 30 or 40,000 individuals, make sure that the vaccine is not giving any side effect or anything like that. So I think similarly, if you develop a drug targeting a particular mutant protein, you have to make sure that the patient can tolerate this and with the minimal side effect. Because already we know that when the patients, cancer patients are undergoing chemotherapy or radiation, they'll have a lot of problems like losing hair and losing weight and other complex problems. But I think that is another major challenge. Even if you identify a mutation, it is not easy to develop a drug for that. So these are all the kind of some of the challenges that we are facing in cancer research. I'm sure, I think the new generation of the scientists, from the colleges like that, you will get inspired with all this information. I hope you will get into cancer research and do something in the future. So these are all a lot of people, it's not, I'm not doing by myself, this is always kind of a collaboration. We work with so many people, with so many institutions, so that we can pool our expertise, and that's why we make all the discoveries. If you see some of the papers, always you have 20 or 30 people in each publications because everyone contributes for a major study. So I think with this, I will stop. I don't want to take much time. If you have any questions, anything like that, you have my email. I think I put it in the first slide. Please feel free to write to me, or you have my WhatsApp number. You can talk to the teacher. Feel free to call me. I'll be happy to talk to anyone. Thank you very much for the time. Thank you for listening.
Thank you, sir. Thank you. Sir, there are some questions.
Go ahead. I'll be happy to answer. Yes.
First question is, sir, what is the treatment method for multiple mutation cancer type for the same individual? Is there any treatment method for multiple mutation cancer type in the same individual?
Yeah, that's what I actually explained very clearly.
Yes,
Say, for example, in lung cancer, there are multiple mutations, but all the mutations are not present in one patient. Okay, each mutation is classifying the patient into distinct subset. So if you take 100 individuals, one particular mutation may be present in 10% of the individual, other mutation will be 5% like that. So it is very rare, very, very rare cases we found more than one mutation in the same patient. So most of the time, every patient is identified with only one particular mutation. Although, say for example, in lung cancer, there are 15 type of mutations are there, you are not going to find all the 15 mutations. You will find either only one mutation or you don't find anything. So actually there is no such thing, although there are so many, like I showed you, like one example, the breast cancer, it has like 80 or 100 chromosomes. Those are all just, it is more what we call actually private mutations, like those translocations are present only in one patient. We don't find the same breakpoints or translocation in the second patient. So that it is very difficult to say what is the role of this mutation in that particular patient. It is hard to develop a drug. Okay, that's why I told you that we need to identify the mutation across several patients, or it has to be recurrent. If it is present, and maybe even if it is 2% of the patients, then it is worth investing our time and effort to develop drug to treat them. So there is no, although there are so many mutations, for most of the mutations, we don't know what is the role and how to actually target those mutations, because most of them are kind of private mutations. Right now, the cancer research, we are selecting only the mutations that are recurrent, meaning that are present across multiple patients, so that we can develop a drug, because that is more common. So when it is present across multiple patients, then we know that it has some relevance, because all the mutations will be exactly the same in all the patients. Then only the patients that are positive with that mutation, we can give the drug to them. Yes. I hope I answered that question.
Yes, sir.
Yeah.
Thank you for your answering, sir. Dear participants, if you have any questions, you can unmute and ask the questions.
Or you can type in the chat box, right?
Yes, chat. RS, you can type in the chat box.
Yeah, you can type in the chat box. If you are feeling shy in talking to me, you can type there.
Sir, good morning, sir.
Good morning, sir. Solunga.
How are you, sir? Sir, it is really a mesmerizing talk, sir. Even it is always you are opening an area for the awareness in cancer. And if someone is looking really for do something in a research on cancer research, you are always opening new avenues in cancer, sir. like you started with the molecular biology and then went on to bring the information on the diagnosis also. So I have few questions with you. One particular thing is regarding the non-coding RNAs. Non-coding RNAs, and it is a kind of thing that it has to be used as an early diagnosis. And what about your opinion on the early diagnosis of cancer with non-coding RNAs?
Like I said, there are about more than 50,000 different non-coding RNAs. And right now, there's a lot of research going on. We are using a technology called next generation sequencing. Using the technology, we will be able to sequence the entire sequence within the cancer sample, irrespective of, as long as the RNA is expressed, we are not selecting any particular RNA. We just, kind of sequence all the RNA transcript that is expressed. Then we are comparing the expression of this non-coding RNA from the tumor cell by taking a normal sample from the same patient, adjacent, to the cancer area. Then we compare, because the non-coding RNAs are also expressed in normal cells as well. Okay, so we have to separate what is kind of more, or what is important, or what is different in the cancer cell, and we identified certain type of non-coding RNAs that are expressed only in cancer cells but not in normal cells. And we identified actually several of them, and there are papers. If anyone is interested, I can send some of the papers that came from our lab as well. We did more in-depth kind of functional studies, what exactly this non-coding RNA is doing. And I think that field is totally new, because the next generation sequencing technology, it came only in the year like 2010, only about 10-year technology. So now we have sequenced so many tumors and we have so much information, and there's a lot of labs are working on this non-coding RNA, and several of the non-coding RNAs have been identified as actually that are associated with a particular type of cancer. So like I said, these non-coding RNAs are not making proteins. There is no way you can treat those. That's a problem. If it is expressing a protein, then you can develop an antibody, you can inactivate that protein. But since this non-coding RNA is just overexpressed, we have no way of actually how to control the expression of this non-coding RNA. But at least we are trying to identify what are the targets, genes that non-coding RNAs are affecting, so that you can indirectly target. Suppose if the non-coding RNA is regulating a protein coding gene, okay, then we can actually develop some antibody or something to target the protein coding gene that is affected by the non-coding RNA. So this field is emerging. There's a lot of research going on, because like I said, more than 90% of the human genome is transcriptionally active, and there's so much complexity, and we are still a long way to go working on that. Yeah.
Thank you so much, sir, because we are now focusing on in our research we are doing in the leukemia samples we are working on, and we started focusing on for long non-coding RNAs, and
Yes.
we got some relationship with microRNA 195 and other things and all there, and we are little confused, and your lectures given me a lot of information. I'm really very happy that always I'm getting impressed. I'm always fascinated towards your thoughts. So it has opened a new avenues. Really thankful to you. Thank you so much, sir.
Thank you.
Thank you, sir.
Thank you.
Your deliberations are thoughtfully decided
One
and
more
carefully.
Miss, one more question is there from the participant. Sir, is there any specific carcinogen for breast cancer, skin cancer, and oral cancer? That's a question from Tyagaraja College, Madurai.
No, I think because there is no specific carcinogen, especially for skin cancer. Like I said, I think the sunlight, the radiation exposure, sunlight is the major cause, especially the white people, since they don't have the melanin on the skin. There is not protection. Because as you see, we don't, like in India, our Asian countries, we always actually work in the field with bare body. We never get skin cancer because we have the melanin that gives the protection. But the white people, since there is no synthesis of melanin on the skin, I think the sunlight what it happens is the sunlight directly affects the DNA, causes the DNA damage. When the DNA is breaking in the cell, there is a process called a DNA repair. It gets repaired most of the time, but sometimes some of the mutations are not get repaired, but they get fixed in the cancer cell, and eventually they develop cancer. So the sunlight is a major kind of carcinogen, I would say, for melanoma. But for breast cancer, because I think mainly the breast cancer and the prostate cancer are the two cancer types that are controlled by hormones. Especially in women, the estrogen is the hormone. Especially, as you know, actually many of the people who are diagnosed with breast cancer, now they actually do what is called the mastectomy. They remove the breast, at the same time they remove the ovary as well, because the ovary is the major source for the estrogen, because that is producing the hormone, because that will, in order for the breast cancer to grow, it needs the hormones. So they have to actually regulate the hormone level. So that's why they give, especially for prostate cancer, the what is called the testosterone is the major the hormone, that is a male androgen hormone that is actually helping the prostate cancer to grow. So that is the main reason for prostate cancer patient, they treat them with what is called anti-androgen therapy. So there are actually drugs that will actually inhibit the testosterone level so that the prostate cancer don't grow. Or sometimes they do what is called the castration. That means they remove the testes. So because that is the one way to remove the hormone. So the especially the prostate and the breast, these are the two cancers that are regulated by hormone. The hormone level imbalance, that is responsible for causing cancer. So you have to regulate the level of those hormones, like actually removing the ovary in the women with breast cancer, and removing testes in male with the prostate cancer, like that. So there is no kind of specific carcinogen that is causing, because especially for breast cancer, there are mutations that are identified, like the gene called BRCA1 and BRCA2. These are the genes that are most commonly kind of undergoing mutations in breast cancer patients, because now they are using that as a biomarker, like I showed you in my slide. They screen the breast cancer samples for estrogen, whether it is positive or negative, then progesterone, is it positive or negative? Then they look for the HER2, there is another gene called HER2 that is amplified, that is on chromosome 17. So the major type of breast cancer, what we call triple negative breast cancer. So it is estrogen negative, progesterone negative, then HER2 negative. So when they are negative for all three biomarkers, that is the most aggressive type of breast cancer. It's called triple negative breast cancer. Suppose if the patients are positive for any of this mutation, there are specific drugs available, kind of what is called olaparib or some drugs. You can give that drug, the patient will get benefit. So I think it's not only the carcinogen. I think now the time is looking for the biomarkers, looking for what are the mutations. Then you can actually help the patients to get the appropriate treatment.
Thank you, sir.
Okay.
Is there any question? Participants, do you have any questions?
Even if you have questions, you can always email me. I'll be happy to answer.
Yes, sir. Yes, thank you, sir. Thank you, sir.
Sir, you are a hands-on person. Your deliberations brought clear understanding, and you are thoughtfully designed, carefully drafted, actively documented, and beautifully presented. So here is Dr. Mrs. Jemima Florence Borgia, Assistant Professor of Zoology, to propose vote of thanks.
Sir, please. Just stop sharing, sir, please. Stop sharing the screen.
Oh, okay.
Yes, sir. Thank you,
Okay.
sir. Thank you. Thank you, sir.
I deem it a great honor and privilege to propose the vote of thanks on behalf of PG and Research Center of Zoology. First of all, my heartfelt thanks to Almighty God for making today's event a great grand success. I express my profound word of gratitude to Professor Dr. Nallasivam Palanisamy, Associate Professor, Scientist, Urology Institute, Medical Group Urology, Prostate Cancer Research, Detroit, for his kind acceptance as a resource person and for his keynote address on new trends in molecular biomarker discovery for cancer. Sir brought a clear understanding on discovery, cancer biomarkers, cancer prevention, cancer therapy by inhibition of negative immune regulation, advancing precision medicine for prostate cancer through genomics, genomic aberration in lung cancer, current status of cancer therapy, early detection of cancer, next generation sequencing technology. Sir, we are really enlightened with your knowledge and your valuable presence. Thank you, sir, for your crystal clear presentation and top notch. On behalf of the Faculty of Zoology Department, I express my sincere thanks to you, sir. Thank you, sir. My heartfelt thanks to our principal, Reverend Sister Dr. S. Esurani, for her inspiration, motivation in conducting the international webinar and for her felicitation. Thank you, sister. My deep sense of gratitude to our Mother Superior and Secretary, Reverend Sister Dr. B.J. Queensley Jayanti, for her constant guidance, encouragement, and felicitation. Thank you, sister. I am extremely delighted to express my sincere thanks to the faculty members and students from various colleges for their active participation in this webinar. I thank our convener, Dr. V. Shanti, organizing secretaries, Dr. Anita Kannagi and Dr. L. Merlin Diana, and the faculty members of our department for their cooperation in organizing this webinar. Thank you, one and all. Thank you.
Let us move on to the second technical session.
Dr. Palanisamy, thank you very much. Thank you very much.
Thank you. Thank you.
Thank you so much.
Good night. Okay.
Thank
I
you
think I have to go to sleep. Yeah. It is midnight here. It is 1:30. I have to go to sleep. Thank you.
Thank you so much, sir. Because
No problem.
3 hours you have for go, no? You're sleeping us. Good night,
It's
sir.
already 1. Good night. Bye-bye.
Good night, sir. Good night.
Good night, sir.
So I request Dr. Yokesh Kumar from Zoological Survey of India. Sir, please share your screen, sir.
Sir, most welcome. Good afternoon, Kumar sir. Good afternoon from principal.
Thank you, sister. I am delighted to welcome and introduce Dr. Yokesh Kumar, scientist, Marine Aquarium Regional Center, Zoological Survey of India, Ministry of Environment, Forest and Climate Change, Government of India, DICA, West Bengal, for the guest lecture of our 2-day international webinar on recent trends in life sciences. He received his doctoral degree from VOC College, Manomaniyam Sundaranar University, Tirunelveli, on the topic, Study on the Impact of Industrial Pollution on the Coral Reef Islands of Thoothukudi District, Gulf of Mannar, Marine Biosphere Reserve, Southeast Coast of India, and his postdoctoral degree from Zoological Survey of India, Kolkata, on Marine Cnidarian Taxonomy. Sir has valued 2 PhD theses, conducted 1 public viva, submitted 5 sequences in GenBank. He obtained his research experience by holding various positions in Zoological Survey of India, Gujarat, Young Scientist and Administrative in Charge in Zoological Survey of India, Andaman Nicobar Islands. He has been published 38 research articles. Most of the articles were in Scopus Index as well as in Heights Index journals. He has very rich research experience. For his effort and his priceless service, he deserved to receive grants from DST SHARE and Zoological Survey of India, MOFET. In order to add his research experiences, he has completed 3 major projects and he is having 3 ongoing of his credit. He was a man of adventure. To prove his skill, he drew a certificate for Paddy as a dive master. He is an excellent underwater videographer. He is a man of exponential knowledge on present environmental and forecasting fishing information. He served as a participatory rural appraisal training and disaster preparedness planning at coastal villages from Rameshwaram to Tuticorin and Pondicherry coastal villages. Today, we are fortunate enough to have such an eminent scientist amidst us. Let us all ready to lend our ears to listen his lecture on recent trends and innovation from the tropical marine forest. Let me hand over the session to Dr. Yokesh Kumar.
Thank you, madam. Thank you very much. Thank you, Dr. Shanti, Convener, Associate Professor of Zoology, for your warm welcome address to Dr. Yokesh Kumar, Scientist, Government of India, West Bengal. Sir, now it's your time to the second
very good morning. Very good morning to all. So my screen is visible, madam?
No, sir. No, sir.
Okay.
Now it's visible, sir. Visible.
No. It's done.
Yes, screen is visible, sir. Visible.
It's visible, madam?
Yes, sir. Is it visible? It's visible.
So now in the full screen, no? It's showing on full screen.
Okay.
Yes, sir.
You're getting the full screen, madam?
Full screen, sir. Full screen.
Okay, okay. First of all, I thank to this principal of this college and webinar organizing committee members, and especially the madam Santi, madam, Dr. Santi, and frequently connecting us and we give the opportunities to this webinar. I thank you very much, and all the students, participants, faculty members. Thank you one and all, and especially I thank to our director gave this permission, this webinar. Thank you very much. Now we are going to discuss about ZSA role in the recent trend and innovation from the tropical marine forest. It's we're going to discuss about the ZSA role only, not for other things. What we done it in the nation. So ZSA, everyone knows, more than 105 years old organization working for the nation and taxonomical related work, and we are working from the sea to mountain. The different area we cover: mountain ecosystem and estuarine ecosystem, marine ecosystem, island ecosystem, grassland ecosystem, freshwater ecosystem, and desert ecosystem. Also, we covering the national parks. More than 103 national parks we have, and wildlife sanctuaries, 532 wildlife sanctuaries we have, community reserves, 66 community reserves, and 26 conservation reserves we have, and biosphere reserve, 18 biosphere reserves, and 49 tiger reserves. Frequently, our scientists visiting on these sites and exploring more number of species and assessing the status and conservation related activities. And we have 16 regional stations in different places, and particularly marine working on Chennai Marine Biology Regional Station, Andaman Nicobar Regional Station, and Marine Aquarium Region in the West Bengal. So I am from the West Bengal station, and we have a lot of collections from 1784. So you think about this, how many years before the collections and taxonomical collections and specimen maintenance, and we are keeping that sample, and with the support of all this stuff, we are maintaining all these specimens in Calcutta in head office. So based on this, our collections of Indian specimens, we have around 99,230 species we have, and more than 81,000 species available in the land terrestrial ecosystem, and 17,000 species reported from the sea. So this much faunal diversity as per the 2019 data. So updated one is going on to publish. It's under the process. And I am from the Marine Aquarium Regional Station. It's located in the West Bengal, and it's the border of the Orissa. It's called Diga Aquarium Marine Aquarium. So I think it's one of the big marine aquarium in India. We have more than 24 big tanks with very valuable marine animals and faunal diversity, all the things, and fishes. Mainly, we are keeping the marine fishes and mollusks and endangered species also, for the creating the mass awareness to the public members. So minimum per day 5,000 to 10,000 people visiting our aquarium and giving good feedback. And we are actually only one station more interact with the public from our station. You seeing this middle portion is our aquarium. So our station campus was seeing that, and we keeping some of eel fishes and this line fishes in our aquarium tanks. So everyone know there's a major tropical rainforest in India. So you know the major tropical rainforest. Western Gate, it's starting from the southwest Maharashtra to Tamil Nadu. It's covered more than 4 states, and northeast region, and Assam, Nagaland, Manipur, Mizoram, Tirupur, etc. is covering the northeast region, and Andaman Nicobar Islands is a major tropical marine rainforest. But the total global rainforest cover nearly 2% of the Earth's surface, host for more than 50% of fauna and flora. So that's why it's called the rainforest. It's 50% of wild animals is present in this 2% of area. So think about this value of this. Of this, coral reef as it called as a rainforest of the sea. They occupy less than 0.1% of the world ocean surface, but provide more than 25% of marine fauna and flora. That's why it's a marine rainforest of world. So you can see the picture. You can think about how much of faunal diversity in that place. Coral reef, tropical rainforest. This photo, so whatever photo I am sharing on here is not from outside and website. It's full and full taken from Indian water, and our ZSA photos only are sharing on this group. So you can see this group of fishes and sponges, all kind of fauna. That why is called as a marine tropical rainforest. And think about lakhs of n number of fishes in this ecosystem. So it is attract and breeding ground, and is a shelter for the fishes, and nursery grounds is different acts doing for different varieties of fauna. That is called marine tropical rainforest. And now where it is available in Indian water. So you just discuss about its estimated cover in India 5,730 square kilometers, and major reef regions in India is called Andaman Nicobar Islands. Is a good reef ecosystem, and Lakshadweep reef ecosystem, and Gulf of Mannar is connected with Rameshwaram, near to Rameshwaram to Tuticorin. It's a Gulf of Mannar Marine National Park, and biosphere resort is there. More than 21 islands covered in that place, and Gulf of Kutch is located in Gujarat. It's covered 42 islands, and also we explored some reef ecosystem, and it's not near to this Visakhapatnam coast. Recently we explored that. And Malvan coast ecosystem also explored, and good reef fishes and all the good reef ecosystem is there. And Kerala coast reported some corals on that place. And more number of coral species reported from Andaman Nicobar Island, more than 550 species reported from Andaman Nicobar Islands, and followed by Lakshadweep Islands, 22 species reported from Lakshadweep Islands, 117 species reported from Gulf of Mannar Islands, and more than 63 species reported from Gulf of Kutch. So compared to this, all the reef ecosystem is a very less number of reef corals available in Gulf of Kutch. So what is the type of the coral reefs? So 3 types of coral reef you have. It's a fringing reef type and barrier reef type and atoll reef. So it's a 3 type of reefs we have in Indian water. So particularly fringing reef available in Gulf of Mannar and Andaman Nicobar Islands. Barrier reef available in fringing reef available in Gulf of Kutch also. And barrier reef located in Gulf of Mannar Islands, and some of outside Pondicherry and this place. They recently find out some barrier reefs and this thing. And atoll reef in Lakshadweep Islands. So only atoll reef present in the Lakshadweep Islands, and all the islands is covered with reef and with good faunal diversity. So now we want to know what is coral. So first we have discussed about the coral reef. Now what's the coral? So coral is a phylum Nidaria, is a living animal. So class Anthozoa, and different types of corals we have in Indian water. So I keeping on this photos on here is different types of corals on Indian waters I'm showing here. And the foliose type is a hard corals. We are saying normally it's a hard corals and corals. And it's a fire coral or tube coral, like that they're saying. It's was coming under the order Sclerotinias. And soft corals. It's not hard one. It's just a soft like. It's look like coral and it's a soft substance called order Alcinaceae is a soft corals. And also the tube-like structures is called also. It's also called order Alcinaceae. It's called tube corals. And the black corals is the horny corals. We are saying it's the order Antipetherians. It's not reported more numbers in the Indian water. Very eight species only reported in India water. And recently, there is only reported from Andaman Nicobar Island. And recently, find out this some of black corals from Lakshadweep and Pondicherry, as well as in Gulf of Mannar also. And this one is a order Alcinaceae is the sea fan. Before it's called as a sea Garruniaceae. Now it's merged with the order Alcinaceae. It's a sea fans. And everyone know this red color spot, this stones. These astrologically they're using for this astrological purpose. They're using in the ring, and they're saying it's a lot of benefit. It's true, but it's a skeleton of the Garrunites. So the skeleton of the Garrunites is like this kind of skeletons. They make it, polish it, and making these stones. This is a corallum. Is a species name is corallum. It's available in Indian water also. It's have. And not particular species we have in the Melithridae. That species is available in India. Alternate of this species they are using in the and astrological purpose and also in trading purpose. And another important things is blue coral. It's like this also is very valuable for ornamental trade and as well as jewelry thing. It's also plenty of available in Indian water. A type of corals we saw, that is how we are making the types. Based on this growth pattern, we making this giving the type of the corals. You see this coral is a branching type. So that's why we are keeping as a branching coral. This Acropora, we are keeping as a, we know the name. That's why we are keeping that branching coral is a Acropora branching. It's a species of the name Acropora branching, and without Acropora we are mentioning as a coral branching. CP we are mentioning as a coral branching. And coral polioso, it's like a flower, like it's like underwater flower. We are keeping that, and plate-like structures. And you see the Acropora table. It look like one person sitting on the table. So it's a Acropora table coral, and this coral is a coral encrusting. Its coral growth over this. What kind of structure is available? It's over the growth over that, and that same structure of the platform. So it's called the coral encrusting. 2 types of coral encrusting available in there. And different sets of corals we are showing here also in Acropora branching and Ocelophora. These are like mushroom coral. This is, you can see the mushroom coral. You are saying mushroom coral, this one. And coral encrusting coral, this one. This is also soft coral. Different type of soft coral, different kind of faunas available in this reef ecosystem. And sponges, you can see the barrel sponges is there, and sea lily we are saying is cranites, and fishes go, and bivalves and dead corals. We are keeping some of coding for underwater survey time. We are coding that. We are making this line tape for assessing the data. So the dead coral reef area survey. And sea fans, what I told is a Gorgonid and Alcinaceae before that. Same thing I'm sharing here. There are different type of sea plants we have. More than 520 species reported from Indian water, the soft corals, and this major group is a sea fan is there. And this sea fan exported banned during this our schedule 1986 and as per the schedule on animal, but is export more than tons from Indian water during as per the MBDA report is more than tons exported to other countries is like plant material. So not is a plant material, it's also living animals. See there's a forest. So for that's why we are saying as a tropical marine forest. So good faunal diversity available in that place, and it's also supporting for the reef fishes and other faunal diversity, and it's mostly available in the deeper water. So you think about this, before what we saw this kind of corals, everything in the shallow up to 15 to 20 meters there. So it will stop, the sea plants will start from the 15 to 40 meters. It will densely it is available and it will supporting for this faunal, other faunal. It's a second major group in the reef ecosystem. And sea pens, it's also available in Indian water. You can see pencil, we are saying sea pencil, and feather, this thing like that we are saying this name. And this is black coral, what we are called horny coral. It's like whip coral. It's commonly whip-like structures, and it's called whip coral also, but it's a black coral. So before they only horny coral they reported, and not as a particular species. Recently we find out these photos, everything from Andaman Nicobar Island. You think about the different colors and this thing. Why it's a colorful? This animal is full of color. That's also one question everyone can rise in the mind. So coral actually is not color, but it's associated with one microalgae. It's called as zoantharin. It's associated with this coral skeleton, and during daytime it's making photosynthesis, and this energy will sharing with both. And nighttime the polyps will exposed and capturing this microorganism and feeding that. As you can see the clear picture of the polyps, and each animal is a single animal, single polyps is showing that. And also this corals is supporting for the small crustaceans, all the small shrimp. What you are seeing, the first one, it's a black coral associated shrimp. So the black coral, what the color of the structure, it will change the color of the animal. So it's a mimic that color with this platform and association with that. And this coral is a bubble coral. It's associated of small shrimp inside that. It's mostly you can, if you need, you want to do this study, you must to go and collect this sample from this particular species. It is not associated with other species. Like that, some of crustacean associated on particular group only. And this is only this associated only this black corals. And here down you can see some of shrimp. It's there. It's a see anemone associated one. This is the first one, and the red one is a starfish associated. If you are seeing, if you are in the coastal, if you are going there, if you are going to the intertidal area in Andaman Nicobar, you can see the cushion sponges. It is mostly associated with cushion sponges only, and plenty of available in Indian water. In Gulf of Mannar also we reported the species, and some mollusk and species also available on that and attached with the coral and only available on that same ecosystem. And live coral, why you are saying we are discussing more about this live coral and this forest? So live coral, you think about this, how many species of fishes is available on that place? So for that only the people discussing about this, and they are speaking about this animal, and they are spending more amount for conservation of this. And not only that, and also it is attract the public, and this supporting for the ecotourism also. Though people going the scuba diving is due to that, and very attract to the public, so it is very supportive. I think this is a single colony, you can count it how many species is there. One single colony of coral, you see that single colony of coral, how many number of species attached about that. Then you can think about the coral reef. So that much reef, it will support more than 200 number of example one single coral. So it will support for the very good for the fishes and other associated fauna diversity. So this fish diversity maximum reported for, as per the fish space 2014, 32,100 species reported from worldwide, and around 2,546 species reported in Indian water, and Andaman Nicobar Island 1,434 species reported for Andaman Nicobar, and 725 species reported from Lakshadweep Islands, 1,182 species reported from the Gulf of Mannar Islands, 402 species reported from Gulf of Kutch, and 250 species reported in estuarine ecosystem, and 217 species reported from lagoon, and 341 species reported from mangrove ecosystem, as per the Venkatraman et al. 2015. And reef fish diversity in India. Not before we discuss as a total number of fish diversity. Now I am discussing this reef fishes. Around 454 species and 38 species families reported as a reef fishes in Indian water, as per Anil Mahapatra 2016 report. And of this, damsel fishes are around 52 species reported. So Bomus centridae, butterfly fishes 32 species, sweet lips fishes 16 species, angel fishes 21 species, and parrotfish 14 species, snapper 42 species, wrasse fishes 53 species, grouper fishes 43, sudgeon fishes 18 species, and krill fishes 26 species. Like that, is a lot of aquarium related fishes, reef fishes we reported, and 2016 is reported. These are some aquarium fishes I am showing on here. It's following slides also I am showing this whatever this availability of our resource in India I am showing on here. And tiger fishes on this photo is taken from Andaman Nicobar Island, parrot fishes from Lakshadweep Island, this one, and this is from Gulf of Mannar, and this is also Gulf of Mannar downpour. This everything is reef fishes available in Indian ecosystem, and you can see this much. The slide is bigger, but you can think about only 2 inches of blended fishes. So like that, small organisms, you can see small holes in this place, small holes. These are sponges. It's attached over the sponges. The sponges will attach in the reef ecosystem. So that is the association of this connectivity of this faunal ecosystem. You can see that. And the feeding of again that state is fishes, wrasse fishes is feeding, and the coral surface algae. Also, it's a cleaning of this cleaning activities, like that we are saying, and it's a feeding of that. And sturgeon fishes, you can see different varieties of sturgeon fishes. And recently, this blue species, actually like this Nebdil 3 of they using for the aquarium trade. So recently available in Chennai market. If you are going to Kolattur, you can get it this aquarium market, this one. And banner fishes, different type of banner fishes, and Chaetodonidae is commonly called as Chaetodonidae. And down it's called the black fishes. And you can see the color. This color is attract to the public, and for that only they're trying to keeping in the aquarium. And nowadays, what they're doing is they try to culturing that animal. So Chaetodonidae is you can see different. It's a butterfly fishes. Commonly we are saying the butterfly fishes. You plenty of available in the reef ecosystem, and different this color is attracting that, and they're trying to culturing in the lab. Still now it's not success. That's one negative things I'm sharing on here. But we have this much diversity in India, and very less number quantity only exporting from Indian water. The Siganidae, Bomacandridae, Bomacandridae, and blue damsels, yellow color damsel, a different type of damsels is there, and yellow fin damsel is there, and blue ring angels. It's angel fishes, blue ring angels, like that. Bomacandridae, and goby fishes. You can see one interesting thing of this animal. This goby. It will burrow inside this sand. It will come out, and you can see this last picture. You can small shrimp is there. So what will the cleanest shrimp will come out and clean the way of this goby shrimp head. So it will come out and take the fish, take a feed and go inside. And this sharing of symbiotic relationship will clean the track of this way, and it's collection food. It's taking sharing both and taking food. So you can different colors is photos from Andaman Nicobar Islands. Sorry, it's from Gulf of Mannar Islands and Labrador. So these are wrasse fishes, different type of wrasse fishes. This is available in the aquarium trade and moon wrasse. It is a moon wrasse and cleaner wrasse. This is a third one. It's a cleaner wrasse. It's an interesting thing during this our diving time. So whenever we are going to diving, just you can without movement, if you are standing there, this coming and cleaning your hands and this dust cleaner like that. You think about this standing with fishes, big fishes. The cleaning is mouth. It's over that. It's the third one, right? It's the third one. So it's very interesting things, and there are some symbiotic relationships. Cleaning and taking feed of it's going inside the mouth and cleaning and coming out like that, doing that. And this is economically very valuable. This group of fishes, and more than 2,000 kilo per kg is exporting from Indian water. It's also available, and this one they are using for aquarium trade. This second one, and the down for the blue line damsel, group of fishes is friendly of available in Gulf of Mannar and other ecosystem also, and dotted things is available in all the reef ecosystem and only in the available in the reef ecosystem. And this clownfishes, there's a Bomus entridae, it's a clownfish. So you are seeing and the different types of clownfishes in there. It's a association with sea anemones. So without sea anemone, it's not possible to live. For that, it's a symbiotic relationship is sitting over there. And this only fish more successfully breeded in India, particularly Dr. Ajit Kumar sir, spent it in Annamalai University. He did it this one, and after that he now presently is working in NBFGR, and they are doing this culturing things, and they transfer the technology to the public. So whatever the scientific things you want to transfer to the technology you want to transfer to the public, and this make the livelihood of them. No, this one kind of thing is a supporting for the conservation of the reef. No need to damage the reef. Once if you are going to the reef, you are making small damages, and the reef is entirely destroyed. For that, the culturing all the things is very important. So they done some of team they're doing that one, and you see everyone now that see horse is coming on the scheduled animal, and pipe fishes also there. When we are diving in Gulf of Mannar, we touched and just said to buy and came back. So it's also associated with the soft corals and corals only, coral reef ecosystem. And this animal also the cultured. I think it's done it from Dr. Murugan. He did this breeding activities, this thing. Sea Ranching also done it through the forest department, and from CM Foray and Annamalai University, some collaborative project they done it in Gulf of Mannar. And this coral ecosystem, what you are saw, this much reef ecosystem will last this ecosystem, what will happen? That is another thing. So the coral bleaching. So you are seeing before everything is colorful. What you are saw before all the corals in the colorful one. Now it's lost as algae. What I said before, it's a coral associated algae associated with that. If lost the animal, you can see the fish diversity. Very less, 1 or 2, 3. Enable only you can able to count it that only 4 or 5 species available in one single colony. If the colony will die, what will happen next? So there's no fishes. Same area, same. You can see that the entire reef is dead, and this no fishes and other associated fauna. The linkages of the ecosystem will break. So for this dead reef ecosystem, it will not support for the. It's also supporting. It is a platform for the new recruitment, but it's not supporting for living things. So presently, so some of dead rattles we observed in Gulf of Mannar due to these fishing activities by trawl fishing is going on there. They are making this this kind of thing. And what are the limiting factors? For the coral reef? Naturally, tsunami, earthquake, cyclone, storm, sedimentation, changing in the salinity, increasing temperature, increasing UV exposure, tidal variation, wave actions. So everyone know that tsunami action in 2004 in India. So you think about in the Tamil Nadu coast, in the Nagapattinam, Kanyakumari, this area, we got a lot of damage and death. And you can think about the Tuticorin to Rameshwaram. We didn't get any, its water level only increased and go down. Due to that, only thing is a reef. This is a coral reef islands. It's a border full of this is 21 islands, making us full of reef structures. Is the waves stopped on that place and only the visible that. And it's also a stop. It's a supporting for the cyclone effect. All the things. Not only coral, it's a mangrove ecosystem. They're reported in Ichavaram, near to this Sidamaram, this area. That also then that small portion is not getting any effect, no death rate reported in that place during the tsunami. It's also very beneficial to marine conservation things. And the temperature is we cannot able to stop that, but we can do. So if you are stopped using AC and all other carbon dioxide thing, protection things, you can stop that and reduce that. You can reduce the temperature level and conserve our this. Indirectly, you can conserve our ecosystem. Whatever directly human-made impact and overfishing. Before you can see one small village, only very small people only working on this fish ecosystem. But you nowadays you can go one small place, more than 100 boats standing on there. So over harvesting it will affect the ecosystem, and coral mining now entirely stopped in Indian water, India, due to this schedule, and government take necessary action to stop action of this coral. So it's entirely stopped. And before this, if you are visited in Tuticorin to Gulf of Mannar, most of the house constructed based on these corals. They are commonly saying it's an oracle. In Tamil we are saying it's an oracle or floating stone. Rameshwaram, the people selling as in floating stones. So this everything is available in India, and particularly in the major reef ecosystem area, it is destroyed due to our human activities. Before we are not aware about that. That's why we lost all this ecosystem, and dredging and blasting. Lots of dredging and blasting activities going on, and sewage pollution activities, oil pollutions, and ship traffic. So nowadays this need is lot of need. So we exporting and importing things where for our need purpose. Due to that also the ship traffic is more, maritime activities and ship breaking yards. So the ship is coming out production more. This breaking yard also increase. And also tourism activities. The tourism also affecting this reef ecosystem. You know that a lot of artificial reef in Singapore, they make it the palm tree islands. Like that, Maldives and all the countries, especially Maldives and Thailand, the major income is through this coral reef. It's modern trade we observed in Gulf of Mannar and our ecosystem, over harvesting and trawl fishing activities in Gulf of Mannar, and dumping of this mollusk, all the things. You can see the second photos. That much mollusk they're collecting through the trawl fish and just simply throwing in the shore. If you are replacing inside, that's also support for the reef. It is also made up of calcium carbonate, and it will supporting for the reformation. Also new coral babies want to settle over that. It's a coral recruitment platform. So just we are making awareness to the public and conducting awareness forum and discussing with these local trawl fishing peoples, and we are suggested them to avoid the reef ecosystem area. And our side we are doing this lot of dumping activities of near the coastal. You see this, and this is a recent photo. The lot of mask is there. This is due to COVID. The people using the mask and throwing in near the shore. It's observed in our coast, in Diga coast. We took this photo, and also this nets and anchoring. What they're doing is a small boat. People they're going to fishing near to the reef ecosystem area. So what they're doing, they're keeping the anchor over the reef. So they have small length coral and small boat, small anchor. They're keeping that. it is affect the reef ecosystem. For that, we are saying to avoid that anchoring over the coral. Just avoid that and keep it in some anchor inside, permanent anchor outside the that, and make it some boyas. And whenever you are going to fishing, and make it tied with boat tied with the boyas, and tie the fishing and do the fishing all that. So this kind of thing, and also destructive fishing activities we observed in poisonous fishing activities in Gujarat, particularly in the Meetapur area. We observed that poisonous fishing, not in the Meetapur, it's outside the Meetapur, that Meetapur city. It's Aramada, backside of the Aramada village. Early morning, the people coming there and just throwing some bleaching powders and this thing, and they collecting fishes and selling there. So the people are not traditional fishermen. They are only purpose of money lenders, not for fishermen. So traditional fishermen not doing this kind of work. Based on our observation during last 20 years servicing with them. So very good. The original fishermen, they're not destroying that ecosystem. Whatever things needed, they will collect it and remaining things, throw it inside the sea. They come out from the shore. But nowadays, whatever they're collecting, they brought it to side and selling fishes to fish market, and the remaining things is selling, and for the calcium purpose, the mollusk, everything they're dumping here, and they're selling to calcium cement factory, all the things, and also they're collecting all other unwanted small fishes, all other things they're sending to the poultry for the poultry feeding preparation. So this kind of destructive fishing activities going on in India also. At present, we are making with continuous training with them, and MBDA they're doing net fish is there. They're making continuous training to the fishermen and avoiding this thing, this kind of thing to conserve the reef ecosystem. And this is what the benefit of the reef. Already I showed all the benefit of this, but you want to know that. So major thing everyone knows is of fishing. So a lot of fishing in collecting the fishes we getting from there, or it is connected with that. So this much big fishes, they are coming inside for breeding and just egg laying and going to the deeper place. So egg will grow up there, and that's like a nursery. Just it grow up and it is moving from the reef to deep ecosystem. So like for that is very important, beneficial for the fish breeding, all the things, and scuba diving and recreational activities. Already I told you some Maldives and Thailand, and they're doing this recreational activities, scuba diving and sea walk, all the things. Nowadays in India, a lot of scuba divers started, and particularly Andaman Nicobar Island, in particular island Havelock, we have a lot of dive centers now. A lot of dive centers, they're conducting diving courses, and they're full benefited. If you are once visited this Havelock Island, you know that, and that much fish diversity available in the reef. For that, the people going to that place. And this photo, what I am keeping the backside photo, is taken from the Havelock Island only. Dixon Pinnacle is a place of the reef, is a Dixon Pinnacle, and also it's supporting, and its reef ecosystem will support for sea erosion. So whatever the major waves, whatever this is, it will catching that, and very slow. It will not affect our shore. Like that, it's a barrier of the our shore. Like that, it is supporting our life, and it's a supported the tsunami time. It's a lot of support, and for that now recently a lot of innovation things is going on, and reef restoration activities I discussing in the coming slides, and aquarium trade. So nowadays this aquarium trade also increased, but only 3% of reef fishes exported from Indian water compared to the world market, only 3%. But we have this much resource, but it's a wild collection, entirely not entirely banned. Only the entering inside the reef area, it's a ban. So what they are doing is they keeping the cages in outside the reef area and collecting the fishes and selling to the market. Particularly Gulf of Mannar, easily they're doing that, and some of village people and they're spending a lot of time for that. They're collecting these reef fishes and selling now. So this we need some very good collaboration and some awareness thing. Then only you can improve this business and livelihood activities to the public. And medicinal value Already we discussed in the first session, full of the cancer related things, very interesting things, but it's a lot of anti-cancer activities extracted from this marine fauna, particularly sponges and corals, soft corals, lot of anti-cancer, antibiotic, and lot of things. It's not only particular things. Anti-cancer, antibiotic, antibacterial, antiviral, lot of things they're doing this, and there's also some steroid hormones extracted from that, and prostaglandin extracted from the carbonates. What you see, the sea fans and trypanoids, and some valuable compounds extracted, chemical compounds extracted from this marine organism, and doing this kind of medicinal and developing activities. And recently ICMRI and innovative collaboration is done its lot of work in drugs from the marine. So they done it from Andaman Nicobar Island. Dr. Vinith Kumar sir and under his team, they done it lot of this work going on, still is going on. So during that time, we are also participated for collection and identification thing, taxonomy part, we done it from that. And conservation management, how to conserve this ecosystem, to regular monitoring the effect of chronic stress on the reef community. So we want to regular monitoring is must to conserve this ecosystem, and coral transplantation and restoration. So if destroyed the reef, so due to natural and man-made, whatever things, but we want to restore the ecosystem or transplantation the ecosystem. Also, we can support this artificial reef ecosystem. If artificial reef breeding stock improvement structure. So if you are keeping this artificial reef structure, it will support for the reef fishes. It will attract the reef fishes, not only attract the reef fishes. It's a platform for the settlement of the corals and all associated faunal diversity. And one important thing, it will avoid the reef damage. If you are deploying a lot of to regulate the recreational and tourism activities. A lot of tourism industries this develop in not only now Andaman. So Andaman is a major thing, and now Lakshadweep they have a lot of dive centers now came, and some restriction is there. Lakshadweep admission will without permission we cannot enter inside the Lakshadweep Islands. Like that, some restriction they're keeping there, and Goa they have the dive centers now. Not before there, and recently Pondicherry there have dive centers, Chennai, and recently in Visakhapatnam we have one dive centers. They developed and they're conducting diving activities. Before going to diving, why I am saying is that before going to diving, we must to teach them how to, without disturbing, how to do the diving. If the divers is going inside, so you are going to touch the, you can try it. If you are interested, you can try it. If one small finger you can keep it over this coral, you just remove it and see that. If that small portion will whiten. You think it. If you are going inside, the polish entirely going inside, some stress. It will recover. It will recover. That's not issue. But you are disturbing that ecosystem. That's an indication that. So please avoiding touching the animals if you are divers. Just go and do the diving, enjoy the life, and come back. And also fishing activities, regulate the fishing activities, and avoiding this trawl kind of fishing things. And if you want to do trawling, allow them to deeper water. That is some conservation point of view. It is very important. And what JAC is doing for that? So this kind of thing, we want to need a diversity assessment. So what we are doing, continuously we are visiting this marine national park area and reef area. So Gulf of Mannar, Gulf of Kutch, and Lakshadweep, Andaman Nicobar Islands. Our team will regularly visiting on that place and doing continuous research and collecting data. You can see the first photo. The LIT I mentioned here, line intercept transect method. Myself, we are collecting data of coral diversity and that reef ecosystem. It's taken from the Havelock Island, and visual assessment. So everywhere we are not able to do it with the line transect method. So sometimes there's a coral diversity is very less. So we are not perform this line transect method. So what we'll do this, we can spend it 1 hour or 45 minutes as a visual assessing method. So we can travel to 1 hour distance, we can cover whatever things available in there, we can note it and document it and come back. So based on this diversity assessment, we are doing that. And also underwater quadrate methods. This is a photo quadrate with quadrate and making the photos. It's a one dive instructor, Mr. Vikas from the before he was from Barakuda dive center from Goa. Presently he is from Dive India in Havla. So myself, we are doing some coral long-term monitoring plot. We fixed in 1 square meter long-term monitoring plots in Andaman Nicobar Island. We monitoring regularly and making some growth and ecological changes activities. Also, we are doing this underwater photograph for the display of this awareness creation presentation, all the things, and also we reporting to the ministry what the resource available on Indian water. For that, we are doing this underwater photographical things and videograph also. And some of place we cannot able to do the diving. So we dived up to Visakhapatnam to Gujarat, but above that, in Orissa and West Bengal, it is not possible to do it diving due to water clarity and very less known people available in the scuba diving related and reef related people. For that, what we'll do in this, we are visiting the regularly landing centers and collecting the information of the landing data. This everything is reef fishes, but available from the Orissa and West Bengal coast. You see Lujanide fishes here, the banded scaridae fishes and grouper fishes and say emperor's fishes. It's and wrasse fishes also there, and sweet lip fishes like that. This all the reef associated fishes.
So, your voice is not clear, sir. Sir, your voice is not clear.
Okay, it's okay. Madam?
Yes, now it's okay, sir. Now it's clear, sir.
Okay, no.
Yes, sir, yes.
Okay. There's a third one is a Grand Island shipwreck. It's present in the Goa, near to Grand Island. So Swiss wreck. It's called as a Swiss wreck. And if you dived in Goa, if you are going Barakuda dive center, there's one of major dive site at that place. It is present 12 to 15 meters depth and northwest shipwreck. It's available in Andaman Nicobar Island, South Andaman. If who are visited the North Reef area, there is North Reef. The shipwreck is there, 9 meters depth. And the Japan shipwreck, it's a 28 meters depth. So it is present in the Nicobar. We dived in that place and it's starting from 28. I'm showing you here only 28 meter depth. It is starting from 28, is going deeper. So we reached up to 28 to 30. So after that, this water current is very heavy. So we cannot perform it full that place. We came. And the Peace shipwreck is near to Peel Island, is opposite to Havelock. It's also present in the 9 meters depth. This everything is a tourism site. What I told is a recreational scuba diving activities. All this island, the shipwrecks is acting as a recreational sites. Also, you can see the faunal diversity of this ecosystem. So what you are seeing is the Wember shipwreck. It's exposed to some of portion on this place, and it's 9 meters up to 6 to 9 meters depth is there. What you are seeing, this photo is taken in 2009. Now this portion full of damaged, exposed site you cannot see. It's full of submerged due to sea level rise, or some of people made it some of damage, or they brought it this crushed material, they cut it the iron material like that, maybe. But you can see that there's a lot of fishes, reef fishes associated on that place. The eel fishes is there, banana fishes is there, carp fish is there, bog fish is there, and lion fishes. So a lot of fishes, and also you can see this platform. Coral is there, cup coral is there, and they're doing inside some. You can see this called cage catching. They're keeping some kud. In name then Tamil kud. They're keeping his kud like structures for the collection of these reef fishes for aquarium trade. And Northway shipwreck, we observe this kind of fishes, and this bad fishes is there, and butterfly fishes, snappers, banner fishes, and good number of, you can see there's a cement ship. It's plenty of cement package. We saw that it's not the shipwreck, and some sea cliff fishes also there, Bombay Centridae, damsel fishes, and Carnate shipwreck. What we told, you see the group of barracuda fishes. I'm showing on here very slow. We are taking photo from 28 to 22 meters depth. That's why the photo is not that much clear, and water current also there. So that much water clarity. We tried to do it good photography than that place, but unfortunately it's not possible to done it due to water current, all the things. But we reached on surface and get back. One of my friend Titus, Dr. Titus in Chennai MBRC station. Myself and him only reached that site and returned back. And this thing is a Grand Island. So I told Sushi's right. It's in the Goa. So this is a CM for it scientific team. Dr. Anita Mary Madam from Mangalore station, and Sujata from Karwa station. CM for a scientist and their team. And we dived and explored with them. And we collect the data and fish biodiversity data, as well as coral recruitment status, and reported to the ministry, and they reported to their ICRA institution. And Peel shipwreck is a wooden shipwreck, and recently actually this was synced after tsunami only, and recently they're using as a recreation site, but it's a wooden part is going to damage now. So for avoiding purpose, they're making some of boya, some rocky structure keeping on there, and developing as a artificial reef like this, developing on and Havelock Island. And you can see plenty of fishes on that particular one shipwreck. That is not much more 20 meter length, but this much fish diversity on that place. And also Sindapalli Reef shipwreck, we recently dived on that place. Last year only we dived on that place with the dive center support, and we explored some fishes. And under the study going on, that still now is not published, and study is under the process, and the regular monitoring going on. We are assessing the fish status, and as well as the coral interesting coral study and the coral status also going on there. Here also can see there's a grouper fish. Mostly it is available in the reef only. This is a line grouper, blue line grouper, and this interesting thing is electric ray. If it is mostly it is inside the sand only. So only tail portion only visible during the diving time. When we are going inside, we can saw the tail. If you are not aware about this, if you are keeping hand over this, you will get the current shock. What you are doing the current pipe, if you are touching the current line, you are getting the shock. Like that, you can observe that. So this kind of experience is there, and Steenwan, everyone know that Discovery Channel, the person, crocodile person. So he also died due to this stingray. Not only this one, another stingray there. And bat fishes and some legendary fishes also there under the ecosystem. Now the artificial reef ecosystem we told. So this photo from artificial reef from Tuticorin. It's the opposite to Thrace from village. So it is deployed 20 years before. By MS Swaminathan Foundation, due to they need support from our station. We supported for them, and we explored the reef, but we couldn't. I'm just same shade we are scuba diver, but We couldn't, 3 days we dived on that place, we not find the GPS location. What the tradition of them, you can see, we are using GPS, they are given GPS coordination, we are diving on that place, we cannot find it that reef. Then continuously we are doing 2 days, we are trying that, but we have failed that. After that, to the green, they have local skin divers, actually pearl divers. They are doing 30 meters without any scuba kits. They will go down and collect the scales and come back. That people, they supported for us. They came as only first day. He told this location will go. You can stop the anchor it. We stopped the anchoring. Then the people dived on that place. They told this location is here. You will go down. It is not that much distance from our GPS location, but we cannot find it. But the people exactly taken to that place without any GPS and any navigation things. So they have traditional navigation things. So our traditional knowledge is going to loss due to traditional fishermen. Now everyone is a lenders, money lenders. They are not fishermen. They are thinking only money. They are it's not supporting for the reef. So they not only reef conservation point of view, and this artificial reef is supporting for the lot of fish resource and also conserve the natural reef. So this, if you are keeping this kind of structure, what will happen? So whatever the fishing activity is going on there, it will not damage. So if the damage also, it will not affect the natural ecosystem, and it is supporting for the platform for the other attachment of the corals. You see how much fish resource on there, and also the corals attached over there. So interesting coral is the Favidae is there, and some species available on there, and pipe fishes is there, clown fishes is there, bug fishes is there. Like the different varieties of ornamental fishes, also edible fishes. Particularly in the trace from, I told the fishing village, they doing the fishing on around that area. Also, some of deep region, they have cup coral beds outside the marine national park. It's in the marine biosphere reserve area, and they have very good cup coral bed. They have very good fish resource. They're doing fishing activities on that place. So for conservation of point of view, what we did. So small story of this one, I can share it this thing. And coral transplantation study, we done it in Gulf of Kutch, with collaborative with World Bank for support and G Foundation. GEC, Gujarat Ecological Commission is a direct partner with the World Bank, and they funded to us, and we done this work. That I said, take over and successfully done it the coral transplantation in Gulf of Kutch. Particularly, we done it 3 places in Gulf of Kutch, Narara, Piroton, and Mitapur area. Not in the Mitapur, it's Arambra. It's near to Mitapur. So it's Narara is middle of that. In the Gulf, if you are seeing Gulf of Kutch, in the Gulf, it's the end of Piroton Island is coming inside, and middle of this Narara Island, and its mouth of that Mitapur is there. So we done it in 3 places and compare the growth pattern, all the things. And one important things in world level, it's the first time we made it long distance transplantation. Lot of transplantation and restoration study going on in the worldwide, and they done it only same ecosystem or nearest ecosystem, not from that much long. We more than 3,000 kilometers we transferred from Gulf of Mannar to Gulf of Kutch. We transplanted, and very good result we observed, and we published the data, and continuous monitoring and studies going on. And also we used some different kind of structure. What you are seeing here, so triangle-like structure we are seeing on here. First slide is a small holes in here. Why we are using like this in Gulf of Kutch? The tide amplitude is very more, so more current and tidal variation is there, and it is more than kilometers in exposed during low tide time. In high tide time, what will happen is it will bring it lot of sediments load and coming in, dumping inside the Gulf. For avoiding purpose, we are keeping these kind of structures. But the sediment load will not settle over this platform, it will go down. So coral will escape from this sediment impact. So for that we made it this kind of structure. And another thing, in down one, third photo E, so down one is a floating tree. What you do, it's a high tide and low tide is more here, and more than 5 meters depth, variation is there, high tide and low tide. So what will happen is the boya is there, and the tree structure we keeping the coral in the tree. During high tide time, what will happen? The boya will come up in the surface. So from the surface, it will take 1 meter depth. The coral we are keeping there. So no sediment load will over there. It will avoid sediment load and other impact, and we observe this structure lot of seaweed deposits. So during low tide time, of seaweeds it is settled over there, and it is
breaking, sir.
Also,
sir, Dr.
Yes,
Yogesh Kumar,
yes,
your
madam.
voice is breaking.
Yes, madam.
Yes, sir.
Madam, actually, network some issue. Now it's okay, madam.
Sir, your voice is breaking.
That's okay. Now it's okay, madam. Hello.
நான் 2 தடவை உங்களுக்கு கான்டாக்ட் பண்ணிருக்கேன்.
No, sir. No, sir.
No, sir.
Now it's okay.
வெளியில போயிட்டாரோ? சிவா சிவா.
நான் பார்த்து இவ்வளவு நேரம் பேசிட்டு இருந்தாங்க.
Yes, sir. We can hear.
Yes, please.
So shall we wind up at 1:05 before 1:05?
Yes, ma'am. Yes, very good.
Okay, sir.
Yeah, sure.
Okay, thank you. Slide. We didn't see. We are not able to see your slide, sir. Yes.
Once again, I share it.
Yes, sir. Yes, sir.
It's coming, madam.
No, sir. No.
Start sharing, madam. There's some network issue. Some network issues there. For that.
Sir?
Yes, madam. Some network issue. Not sharing now.
Okay, sir. Okay, okay.
Yes, I'm trying. Once again, I'm trying.
Shall we wind up, sir?
Coming, madam?
There is some question, but it is audible. No, sir. Your voice is audible, but only a screen is not visible, sir.
Screen is not there. Okay. Okay.
Yes, sir. Shall we go for discussion then, sir?
Okay, no issue, madam. No issue. There's only few of slide only.
No, sir. You can conclude, no?
Yes, I'll conclude that one, madam. Just we're going to
Yes, sir.
conclude.
Without sharing the slide, you can conclude, no, sir?
Madam, actually, few of things. I just share it and just then you can conclude it. Excuse me, madam.
Sir?
It's okay.
I'm not
Actually,
able.
madam, I share it some of.
Yes,
You are
sir.
getting,
Yes,
madam?
sir. Yes, you can proceed. You can proceed.
Okay. 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 And another one thing we tried in there in India first time slide is not sharing, but I am sharing here. And first solar power biorods. In India, it's the first innovation things we done it from Zoology of India and collaborated with IIT team and international scientists. Thompson Korea scientists came here and we done it in Gujarat. And Dr. Satyanarayana sir, they're leading the team and successfully is going on. Recently, last year only, they deployed that structure inside and making the solar panel. Photo is not visible. Otherwise, I can explain that. So, but this is innovative things done it from, let us say, and some institution Indian water. And also we conducted scuba diving activities, scuba training to the forest department peoples and public for the awareness creation and conservation point of view. Without scuba, we cannot enter inside the coral reef area, except in Gujarat. In Gulf of Kutch, go in but other place without scuba is not possible. We trained the forest department people for scuba diving to collect information for conservation activities purpose, and they're trained and they're doing the conservation active work. And also I thank our and regularly we conducting this awareness program, and I thank our directors involving continuously for these awareness activities. Dr. A.K. Ghosh sir, actually the first slide is not showing it, so you can see our directors, different directors, how they are involved awareness programs. A.K. Ghosh sir is there, JRP Alfred sir, and Dr. Venkatraman sir, and the present director Dr. Kailasandra sir, all of supporters for, and they're involving for awareness creation activities and conservation activities. I thank you everyone, and the management committee and the thank you very much, madam.
Thank you, sir. Thank you.
Thank you.
Sir, there is one question.
Yeah, madam, please.
What is bio-rock reef creation? What is bio-rock reef creation? What is the role of Zoological Survey of India in India and its successful rate?
Actually, madam, actually last year only they deployed this bio-rock in Gulf of Kutch, and success rate now is coral started. Actually, this bio-rock is supporting. Actually, this is solar bio-rock, madam. It's whatever structures you are keeping inside, and the solar reaction is producing mild current. The current, what will happen, anode cathode current will support for the settlement of the calcium over these structures. So calcium will settle over that. The calcium carbonate, the substrate will create. So juvenile will go and attach over that place. Now what we tried in Gulf of Kutch, not myself, I not participated, and Dr. Satyanarayana done it that work, and the team, and they done it just collecting the local samples and tying over that and monitoring the growth. So still now it's not updated thing. There's a growth and this thing, but we expecting this very successful one, and well-made success. In Australia they done it very good work and success rate is good, and India we tried this the first time. So we are going, and some of the NGOs are involved for the oral transpiration in that place, and the bio-rock structures they deployed in Gulf of Kutch also, madam. And WTI Wildlife Trust of India, one of my friend present on this team is Subram. He supported for a lot of coral transplantation time in Gulf of Kutch, and also in Gulf of Mannar and Andaman Nicobar Island. He supported a lot, and I thank to him, and he is present here, and he is worked on that time in the Wildlife Trust of India. They are also done it coral transplantation in Gulf of Kutch from Lakshadweep Island to Gulf of Kutch. Thank you, madam.
Thank you, sir. Thank you. Participants, if there is any question, you can raise your questions by unmuting your audio. Is there any question? Participants?
If the people interested, madam, can they train them? Now the university is conducting scuba diving activities. So if they interested, involve the people.
So there is a question, sir.
Yes, madam.
Is from Subbaraman, sir.
Yeah,
is
please.
any NGOs involving coral transplantation in India? Is there any NGOs involving in coral transplantation in India?
Further than I told before, madam, I said this from he is my friend, and he worked in the WTA, Wildlife Trust of India. They involved in the coral transplantation in Gulf of Kutch, and under his guidance only they done it that place. He is present here, and you know how they done it, and they are also supported for site selection in our study also. I thank to him, and he is present with us.
Thank you, sir. Thank you. Is there any other question from the participants? Participants, please. Is there any other questions? Shall we wind up the session then? Suppose if you are having any question, you can mail to Dr. Yokesh Kumar, sir. He will answer you. If there is any question,
Definitely. The mail ID is there, ma'am.
you can
Mail ID
mail
in the-
directly to sir and he will answer for your questions. Attendance link is posted in the chat box. Please fill and send it back, please. Attendance link is posted in the chat box. Please fill it before you leave. Vote of thanks.
Vote of thanks by Dr. Mrs. Delphine Rose, Associate Professor of Zoology. May I request Dr. and Mrs. Delphine Rose to propose vote of thanks.
Good afternoon to all. I express my sincere thanks to Dr. S. Yogesh Kumar, Scientist, Zoological Survey of India, West Bengal, for his acceptance to serve as the resource person. Sir brought a clear vision about recent trends and innovations from the tropical marine forest. He explained about the major tropical rainforest, types of coral reef fish diversity in India, coral ecosystem, and the conservation and management. We are really enlightened with your lecture, sir. Thank you, sir, for your wonderful presentation. On behalf of the PG and Research Center of Zoology, I express my sincere thanks to you, sir. Thank you, sir. Thank you, one and all. Thank you.
Thank you very much, madam. Thank you very much for your support, and thank you principal and Santi madam also. Thank you very much, madam. Thank you.
The attendance link is posted in the chat box. See, fill the attendance link. Dear participants, the attendance link is in the chat box. Please fill and send it back to us. Sir? இப்போ நான் க்ளோஸ் பண்ணிக்கலாம்லடா உன்னோட
அதையும் குடிச்சிக்கிட்டான் ஆள் போனோம் செய்யறதா கஷ்டம். மட்டன் குழம்பு சிக்கன் கிரேவி நான் செஞ்சேன். ஃபிஷ் இதெல்லாம் செஞ்சுட்டாங்க. மீதி அதெல்லாம் செய்ய முடியாது. சாமி கரையும் போது போட்டு ஏதாவது பாதிக்காங்க. ஆமா பெரிய வேலைங்க. ஆமா விளக்கி கொஞ்சம் வச்சு விளக்கி நல்லா படையல் போட்டு போட்டெல்லாம் எல்லாம் செஞ்சா டைம் ஆகும். அவங்க மாமனார் வந்து அவங்க அம்மா அப்பாவுக்கு சொல்லி கொடுத்து நான் வளர்த்துருந்தாங்க. அந்த மாதிரி உங்களுக்கு திட்டி கொடுக்க மாட்டேன். திட்டி கொடுத்தா நாங்க மாட்டோம். அப்படிலாம் அவங்க பண்ணுவோம். திட்டி கொடுக்குறது திட்டி கொடுக்கறது தான் பேசுறாங்க. திட்டி அன்னைக்கு அதெல்லாம் வீட்டுக்கு வந்து அப்படிதான் வருஷம் நீங்க அப்ப சாரெல்லாம் கொடுத்துருவாரு. நீங்க சார் கூட சொல்லிக்கலாம். ஆனா உங்க மாமியார் கேட்குது அந்த குடும்பத்துக்கு வழக்கம் தான். மாமனார் தெரியல பர்சன்டேஜ் நல்லா இருப்பாங்க. எல்லாத்தையும் கூப்பிட்டு பர்சன்டேஜ் கொடுத்தாங்க. அதுக்கு தெரியலைங்க. நாங்க சொல்ல மாட்டோம். உள்ள கொடுத்து எல்லாம் ஆயுக்கும் செய்யப்படுறாங்க. அப்புறம் அதுனால சொல்றாங்க இருக்கும்போது பார்த்தா இருக்கும்போது நல்லா பார்த்தா தெரியும். அது என்னங்க ஒரு வேலையே கூப்பிட்டு நான் சாப்பாடு தானே வச்சு கிடைப்பாங்க. அதுனா எப்பவும் செய்யற மாதிரி செஞ்சுருப்பாங்க. ஆமா போடுற மாதிரி அவங்க வயசுல பொருள் எல்லாம் கலெக்ஷன் பண்ணணும். அது நாங்களே வாங்கிக்க மாட்டாங்க. சுபத்திருந்தா எக்ஸாம் வருதுதான் கேட்பாங்க. இந்த பொண்ணு பார்க்கவே முடியல. மனது வருது. தேங்க்ஸ். பாப்பா பண்ணுறாங்கப்பா. நன்றி நன்றி.